All right, so I think we're at a critical mass now. Ready to get started? Do I need to stand here so people on Zoom can hear me?
Yeah, you can see yourself on the screen too.
Oh, no. All right. All right, how's that look?
I don't know how to do that.
Well, let me drive. Go to the video set.
Oh, then you should do it, you know.
That work, Casey? All right, so we're running a couple of minutes behind schedule, so we're gonna move quickly through this first couple of minutes. So I wanted to first of all welcome everyone to this year's Shoals Undergraduate Research Groups Research Symposium. We're really psyched to have everyone here. So many of you I learned on the boat are kind of new to Appledore and to the lab. So welcome to all of y'all, particularly a couple of y'all that flew in from very far away. So we're psyched to have everyone here today. A quick background on the lab itself. So the lab is jointly operated by University of New Hampshire and Cornell University. And we're focused almost exclusively on undergraduate teaching and research. We've been doing it for over 55 years, which is something that we're really quite proud of. In addition to the undergraduate researchers that you're going to hear from today, the lab offers a host of undergraduate courses, everything from like the really tiny microscopic parasites to the sort of charismatic megafauna. We have a couple of classes on marine mammals and sharks. So we try to cover sort of the full breadth of marine science here at the lab. And so embedded in all that sort of academic course structure, we have our research interns that are here with us for the majority of the summer. So we've been offering these internship programs for many years. It's something close to 20 years. When it first got started, it was funded through a National Science Foundation grant, and it's since sort of transitioned into something that the lab has focused internally. And we're really grateful for a host of different sources of support that help facilitate the undergraduate research program. That includes the Dartmouth College's Office of Undergraduate Advising and Research. We have 2 students that are with us here today from that program. New Hampshire Sea Grant has been a longtime supporter of the lab, as well as several different funds and endowments that the lab leans into to help support this program, including the Bartels Fund, our annual gift fund, and the Tuttle Fund. So without these individuals and partner organizations, we really wouldn't be able to be here today. So we're really grateful for all of that support. One of the wonderful aspects of the Surge program is that during this sort of 10-week period, all of these students that you're going to hear from today become really integral parts of our island community. And so that's one of my favorite things about them being here. This is sort of the culmination of their 10 weeks. But they're not just doing research. They're here to, participate in food runs. They're helping out in the kitchen. They're even doing like late night taco nights with the kitchen staff. They're having all manner of different aspects of kind of life here on the island. So we're really grateful for their participation in our island community. It's really awesome to have them. I would also be remiss not to mention all of our staff that help make this 10-week program happen. Everything from, y'all that came out with us today, Captain Steve is sort of the leader of our waterfront department. So Steve, along with Captain Heather and Captain Roger, have been facilitating a lot of the research that you're going to learn about today, whether it's dropping underwater cameras or doing surveys around Duck Island or pulling traps for lobsters and crabs. The waterfront department has been really crucial to all of this research that we're going to learn about today. Also our island engineers, they're kind of the secret people behind the scenes that keep the power moving. They keep all the seawater flowing through our sea tables. So without the work of Zach and Ken and Doug, these programs would also really would be faltering. So we're really grateful for their help. Our programs department, which consists of Merrick and Sarah and Kelly and Jenna, are also really integral to making all this happen. Particular shout out to Kelly for all of the work that she does scheduling all of these boat trips that you're going to hear about today. So thank you, Kelly Morgan. And then our kitchen, they're sort of behind the scenes right now. They really they are the engine of this island. They bring all the yummy food, keep everybody fed and happy. So thanks to that staff as well for making all this happen. And then, so before I hand over to our MC of the day, that we also I think we all should give a big shout out to our scientists in residence. Rebecca Atkins and Mike Sigler have been, gosh, they've been really huge to this program this year. Thank you, Rebecca. Thank you, Mike. They've helped guide all these students from, the first week of June up until now, and their full progression of learning how to do research, learning how to talk about research. So we're really incredibly grateful for your support. So, Master of Ceremonies, Rebecca Atkins, please come on up.
Thanks, Dave. So for our undergraduate researchers, today marks the culmination of their 10-week summer here on Appledore. And these passionate scientists arrived in early June, and this is their final weekend with us. Bittersweet. So for many of them, this summer represents their first foray into independently conducted field research. And so as you'll see, each and every one of them, they hit the ground running from day one. They grew substantially in their confidence and abilities as scientists and seeking answers through an iterative, collaborative, and scientific process. Each researcher has conducted an independent project guided by research mentors both on and off island. And these research mentors have provided guidance around study design, field sampling methods, data analysis and interpretation, and the broader ecological context of the Gulf of Maine. And while many of our primary research mentors are not residing on Appledore during the summer, they make 1 to 2 trips to the island throughout the summer. They hold regularly scheduled meetings with the undergraduate researchers, and they have infused our undergraduate research program with abundant enthusiasm and a suite of scientific skills to share. So I would like to take a moment to acknowledge each of our primary mentors that have been so instrumental in the success of our undergraduate research program. And you can find all of them listed on the back of the program that you have on the tables in front of you. And in addition to the primary research mentors, both Mike and I, and our role as scientists in residence, have been dedicated to working closely with this team of researchers on their day-to-day challenges of field-based research. So I can say with much admiration that over the past 10 weeks, our interns have worked tirelessly on a range of research topics that not only are locally relevant and important here within the Isle of Shoals, but also have contributed to larger scale research questions within the Gulf of Maine and globally. And I would like to convey on behalf of both Mike and myself that it has been inspiring and deeply rewarding to work with each and every one of them this summer. And today's symposium represents the culmination of their work here at the lab. The program that you see in front of you, even though the times might be a little off, outlines the full schedule for the day. We will hear 7 presentations from our summer undergraduate researchers before lunch. And then in the period after lunch, we will hear a talk from our summer intertidal research group and students at Shoals Marine Lab's research apprenticeship program. And these young scientists have been asked to prepare 10 to 12-minute talks in the style of a traditional scientific conference. After each talk, there will be time for questions. So I encourage all of y'all to jot down notes and be prepared to ask questions at the end of their talks. Before we start, I also want to highlight the Surge intern group as a really fabulous group of folks. On Appledore Island for 10 weeks, they have created a vibrant and supportive community that extends well beyond themselves and also includes the intertidal long-term research mentoring interns and many waves of student classes that have passed through Shoals Marine Lab at 2-week intervals. And so even in their free time, when they're not frantic in the field collecting their data, they have organized adventures and they have shared their love of painting, music, fishing, and foraging with everyone on the island. And these young scientists presenting their work to you today, I can say without a doubt, are generous, they're creative, they are kind, and they are bringing a wealth of knowledge and enthusiasm to share with you today. They're first going to share a group presentation, which is meant to celebrate this community that they have created here at Shoals and to inject some humor into the start of our symposium. So without further ado, I will hand off the show to them. By that, I mean I will get the tech situated. Mhm. You want me to hit play? Okay.
Okay, I just want to reset the timer. So. Ooh. Oh, okay. Good morning, everybody. Welcome to the Surge Symposium. My name is Madeline Wenger, and I'm going to kick off our talks today with my research about the underneath the surface along the subtidal zone of Appledore Island. Let's get into it. First, I'll be discussing my overarching survey of fisheries diversity and abundance per species, then an alternative bait experiment that I conducted, and finally, the guiding principles and fisheries philosophy that informed my project, and there will also be time for questions afterwards. So this summer, I've been conducting an underwater survey of subtidal diversity and abundance per species using remote underwater videography. Pictured are the PVC camera mounts that I deployed during a 5-week sampling period. This summer, I sampled different sites across the 4 corners of Appledore's coast. Each site has 2 depth categories, fewer than 8 meters and between 8 and 20 meters. And this is the project's 2nd summer. This year, we documented how factors of depth, temperature, and exposure to the open ocean all affect which critters I'll see on camera. Once I collect my cameras, I manually review the footage. I start with environmental observations like what percent of the view is rocky terrain versus algal cover, or just a view of the water column. How long is the vegetation in view, and how aggressive is the sway of the ocean? For example, I would say that this is about 50% rocky or sandy. It has about 20% algal cover, and it includes short, medium, and long algae. Next, I identify as many species as possible in the footage. This is a cunner. It's one of the most common fish that we see around the island, and I typically identify it by its rounded and stout caudal fin, or its tail, and the way that its dorsal and pelvic fins collapse when it's swimming. In comparison, the American pollock is identifiable by its shallow or more tapered body plan. It also has a forked tail, a bright lateral line, and angular fins on its underside. As you can see, it can be a challenge to estimate a fish population from a limited camera screen in such a dynamic environment. So a major question is, how do we count a school of fish? In order to capture a conservative and representative measurement of fish populations, I'll count the maximum number of individuals per species in a single frame of footage. I work under the assumption that fish school together and they're from the same species in a single school. And this is the industry standard for preventing the double counting of fish. I don't actually do this. This is just kind of illustrating how my eye works when I'm reviewing footage. Now let's talk about outcomes. First off, we can refine replicable procedures to build a database about the island's subtidal ecology to accompany our decades-long database from intertidal fieldwork. From this, we can establish the baseline conditions for the Isles of Shoals to learn about, for example, how climate change is affecting our coastline. It's not a perfect reference for diversity and abundance because we are playing catch up, but it is never too late to do this research. Within this historical data set, we can investigate the relationships between environmental conditions and the species we see and the population size per species. To orient you, in this graph, I'm comparing the depth of the underwater camera setup and the number of species viewed in a single piece of footage. And for context, one camera will be about 60 to 80 minutes of footage. Now we can see a general trend of footage collected at deeper depths presenting more diversity or a higher number of species. In this graph, we can see a clear relationship between deeper depths and cooler seafloor temperatures. And this is because cold, salty ocean water is denser and it sinks to the bottom. We can round out this initial glimpse at how temperature, depth, and diversity relate. In this graph, we're looking at how seafloor temperature on the x-axis affects or relates to the number of species on the y. At cooler temperatures, like I said, often deeper camera deployments, there's a greater likelihood of finding more species in the footage. In this data set, we can also continue to get more specific about how depth and the number of individuals per species relate. This graph is specific to the Atlantic pollock. And something I've learned this summer is that pollock are found in groups or even schools at progressively deeper depths. But as you can see, we can actually mix and match the different variables that I've collected data on, and we can learn about subtidal ecology at a species level. I'd also like to take an opportunity to illustrate where I plan to take this project in the future. I'm only beginning to learn about these concepts that are illustrated here, but at a basic level, this graph demonstrates co-occurrence of species. It draws upon the equivalent of dozens of comparisons between species to narrow down which species tend to be found together in camera footage. The data shows in the top right corner that cunner and pollock are commonly observed in the same footage and environment. In the bottom left, there's a similar relationship between lobster and crab appearing at the same time. The next step would be superimposing this information about co-occurrence over the geography of Appledore Island, and we could learn about which species live together and where. Now let's transition to the alternative bait experiment I designed, and I'm using the same underwater camera technology as in the previous survey. For context, there's an ongoing migration of invasive species into the Gulf of Maine as it warms at an unprecedented rate. And particularly destructive and disruptive is the European green crab, Carcinus manus. We have local stakeholders like Dr. Gabriella Bratt, who wants to intervene, and she actually came to Appledore in June to discuss introducing green crabs into local Portsmouth seafood restaurants. At the same time, herring and mackerel fisheries are overfished and becoming more and more expensive to source from. And so this leads me to researching green crab as an alternative bait resource. My main research interests are how can we pivot away from overconsumption of bait fish? And what's key to note is that right now fishermen are using about 2 pounds of bait fish for every 1 pound of lobster that they catch. In response to this, can we use a plentiful resource that is disrupting the local subtidal ecology that I'm already monitoring? So what I did is I baited my camera rigs with 3 different treatments. I used green crab by itself, I used mackerel by itself, and then an even mixture of the two. I sampled 6 sites that were shallow in depth, testing for the number of times that a critter, including fish or lobster, would tap the bait bag. Now for what I learned. My data ended up having so many videos without bait interactions that it's hard to effectively depict a pattern of bait preference. However, green crab consistently marked 0 bait taps. Comparatively, the bait engagements that I was able to document were baited with mackerel or mixed bait. And this tells me that we should shift gears in how we approach this research question about responsible bait usage. I would recommend a field experiment that baits lobster pots using progressively lower ratios of mackerel to filler baits that could include green crab. And this would illuminate a threshold for the minimum possible of mackerel needed to fish successfully. Let's take a quick brain break and look at some highlights from this summer. So this is Connor. This is the fish that I said was one of the most common. These are white flounder clips. It tends to hang out at the bottom. It's a flat fish. It seems to prefer sandy and deeper water. An ocean pout is another rare species that I only saw once this summer, and it's at 16 meters in sandy environment. This is a silver hake school. It's identifiable by a very tapered body plan, and it's very reflective. We saw a lot of striped bass down at the southwest side of the island. Some Jonah crab footage, which will be relevant for a later presentation. Bottom left, you can see a rock gunnel kind of sliding through. And in the top left, you see a red sculpin moving around. And there's also overlap with other projects, including the molting and vibrissae projects. As well as occasionally there's a surprise seabird on camera. And then this is an example of what the baited interactions look like. These are with mackerel and mixed mackerel and green crab baits. So now that you've learned about how I went about collecting data, I want to highlight the guiding principles of fishery science that inspired and directed my research design. For this metaphor to work, keep in mind that a good boat, for example, the Zodiacs that we depended on all summer, need a good propeller. So if we treat my project like a boat, these are the principles that propelled my research. Zeroing in on local issues, on Appledore, we have a really rich intertidal time series, but our understanding of what controls subtidal diversity is less clear. I also wanted to outline a question that could be useful on the daily to local stewards and stakeholders like lobster fishermen. And to do this, I asked, how can we cut down on bait usage as herring and mackerel prices soar? Researching how we can use invasive green crab to pivot away from overfished bait resources ties in with climate change awareness and promoting sustainable fisheries management. And finally, I wanted to make sure that my research was accessible, digestible, and engaging for the public. One way I did this was to make this interactive plot that you can actually end up access online and see all the data from my camera deployments this summer. And things that I'd like for you to remember after this talk. We are only beginning to understand what the current baseline for subtidal ecology looks like around Appledore. There's promise in decreasing our use of overfished bait like mackerel while still engaging fish and lobster. And it's crucial to design questions in partnership with the people who are most affected by the local issue in order to make an impact. These are my references, and fun fact, all the illustrations in this presentation are original, so you can access my portfolio there. Thank you so much to everyone who lent a helping hand and mentored me during this incredible process. Thank you to Dr. Rebecca Atkins, Dr. Mike Sigler, Dr. Jen Dykstra, Dr. Easton White, all of the island staff in the kitchen, down at the waterfront, and in the grass lab. Most importantly, thank you so much to my fellow surgeons, Bella, Kieran, Sebastian, Abby, Noah, and Sarah. I couldn't have pulled this off without your support. Thank you. Any questions? I'll be happy to take right now.
Translating the research into advocacy and providing information to lobster fishermen so fishermen start using the mixed data.
Absolutely. So one of the ways that we could go about that is kind of touching base with Dr. Gabriella Bratt, who came here in June and was working on bringing it to restaurants, and talk to her because she's working with people who are farming green crabs. So that would be an easy way to access them for bait. So we wouldn't have to set up a separate fishery. But another option is to get the word about interactive plots like this and show the relationship between how alternative bait can be successful, but that would only be after we do another experiment that I mentioned, where it kind of proves that we can shift the amount of mackerel that we're depending on and also up the amount of green crab that we're using. That's a great question. So my camera setup didn't have an accurate scale. So it's actually really difficult to figure out if it was a juvenile fish versus a fish that's just like very far away versus a massive fish, or if it's just really up close. So that would kind of have to be revisited later.
I was just amazed by the 2 pounds of bait, 2 pounds of bait for 1 pound of lobster.
Right.
So are you suggesting just changing the proportion of bait, or also a reduction in the amount of bait?
I think that both of those would be important to research. So I think that we could afford to cut down how much we're using in general, and then within that ratio, how much would be like an oily fish that's like mackerel or herring.
If we had something wet, I'm wondering if you guys would grasp about temperature and wondering how with all of that, like one of the moving off of the island or something island. Did you see any kind of response, after those big rain?
Yeah, there were days with really strong fog cover, and that meant the visibility underneath the waves was like very bad. So that probably impacted the amount I could count, but also I would expect that the fish are still there. So it's kind of strange that I didn't see them. So I'm not quite sure how to answer that question, but I definitely noticed a change in visibility, which could have affected how many fish I saw. Oh. Oh, yes. Yeah. Yeah. That would be great. I would say that UNH is probably more likely to be the right group to talk with because they are on the local coast. But that would definitely be another project as I transition this back to school. Should I repeat the previous questions that I answered? Okay. Okay. Okay, great. Thanks so much.
We can see the presentation on you. No. All right, well, thank you for coming, everyone. I'm excited to talk about my research project this summer, and Madeline set the stage with it well, talking about the lobster fishery, but my project was focused on the distribution of juvenile American lobsters in the Gulf of Maine, as well as around Appledore Island specifically. So first, to just give some context about myself and how I was interested in this project. I'm from Cape Cod, and I used to commercially fish for lobsters out of Nauset Inlet, and I was also a fisherman in a razor clam pilot program in my town. And this program was geared at seeing the viability of razor clam harvesting in Cape Cod Bay. And I was able to attend management meetings in this town, in my town around this, and it got me inspired to take on a research project like this. So the roadmap for how I did my research this summer was first comparing the surveys used that contribute to the East Coast American lobster stock assessment, and try to take on a novel perspective on the stock assessment in that sense. And then secondarily, I looked into the juvenile lobster distribution around Appledore Island with my own field survey out here. So some background about the lobster fishery and the Gulf of Maine. As many of you know, it's one of the quickest warming bodies of water in the world. And this is because of the Atlantic meridional overturning circulation that's slowing, and the Gulf Stream is a certain section of this. And as the Gulf Stream slows, it moves northward, pouring warm water into the Gulf of Maine, as well as pinching the Labrador Current off the southern coast of Nova Scotia. So this is part of why it's warming here so quickly. And when this area has the most valuable fishery in the United States, this can have serious economic, cultural, and community-like implications. So using juvenile lobsters as a way to forecast what's going to happen in the fishery in the future can be an interesting way to look at this issue. So the lobster fishery is a federally managed fishery, and over to the left you can see the management zones across the East Coast. We're in Area 1, which is the light blue furthest north, and it spans across the coast of Maine all the way into Cape Cod Bay. And these are the state management areas where the Isle of Shoals lies in Area G. Here is some landing context comparing the southern New England and the mid-Atlantic to Maine and New Hampshire landings. And in the early 2000s, you can see there's a precipitous drop off in catch in southern New England and the mid-Atlantic, while catch was booming in Maine and New Hampshire. And much of this is attributed to environmental pressures, although there's some debate over how management tactics in these two different areas had an effect on this as well, as Maine tended to be more progressive with how they notched lobsters and put eggers back in the water. Here's some effort context in the state of Maine, which is just the total volume of lobster traps in the state. And you can see it kind of starts to approach stabilizing around 1990, and that's because of some legislation that created an 800 pot limit for fishermen. So this effort, it's tended to stay consistent in the last 20 years. And comparing the landing data to the effort data, I can calculate catch per unit effort, which is an important concept I'm going to get into very soon. Here is the southern New England and mid-Atlantic landing data compared to the average sea surface temperatures in the month of June, July, and August. And I picked these months specifically because they're the highest landings for lobster within the year, and slightly bleeding into September as well. But these months are also some of the warmest sea surface temperatures, and those upper limits of temperature throughout the year can have the most influence on the migratory patterns of the lobsters. And then this next plot is highlighting the catch per unit effort of lobsters in Maine. So it's, as opposed to the landing data, I'm standardizing the catch over, often catch per unit efforts calculated the landings or the pounds of catch over the time spent fishing. But with many of the surveys and the work I did, it's over the gear that was used. So in this case, it's the pounds per lobster pot. And yeah, moving on, this is the lobster life cycle, starting with mating. The females will carry eggs for up to 9 months. The eggs will hatch and the larvae will go into the water column, feeding and eventually settling to the bottom. And then within the first 7 years of their life, they'll grow to the legal harvesting size of 3 1/4 inches on average, and they'll molt around 25 times during this period. And when I refer to juvenile lobster, I'm just referring to a sublegal lobster being under 3 1/4 inches. And in Maine, there's also an upper limit of 5 inches from harvesting to try to preserve the mating and breeding lobsters. So part one is stock assessment analysis. The stock assessment is broken into 3 major surveys conducted by the Division of Marine Resources, being the Inshore Trawl Survey, the American Lobster Settlement Index Survey, and the Ventless Trap Survey. So here I'll describe these surveys. The Inshore Trawl Survey is broken into 5 regions across the state, as well as different depth strata, and it's done 2 times per year in the spring and the fall. And this is just a trawl behind a boat, so it doesn't select as well for lobsters as other surveys might. So it contributes to the stock assessment of many species outside of just lobsters. Here's the catch per unit effort of the inshore trawl survey over the last 20 years. It shows the proportion of juveniles to adults that tended to stay consistent over time. And this is indicative of a healthy age structure in the fishery. And the one like anomalous data point I'd say is 2020, and that's because there was only a fall tow because of COVID in the spring. So if you were to roughly double the magnitude, if there was a spring haul, it'd follow this pattern, seeing a slight decrease in recent years with catch per unit effort peaking at around 2015. Now this is that same data broken down further by region and seasonally by the tow. And the biggest takeaway from this is the northern regions, being region 4 and 5, have shown more fluctuations in catch per unit effort than further south, where it seemed to be more of a maintenance of catch per unit effort. And this could possibly suggest expansion of lobsters settlement regions over time, the more northward regions and areas. Next is the American Lobster Settlement Index Survey, which is a diving transect survey, and they also use a suction tube to collect the larval lobsters. And so yes, this targets the smaller lobsters and even can collect like young of year lobsters that are often hard to see with the naked eye or catch with nets and traps. This is the catch per unit effort, in this case being the number of juveniles that are in per quadrat. And this has a lot greater noise than the catch per unit effort of the trawl. And it also, this plot indicates more stable juvenile settlement than the trawl suggests, where we're seeing landings start to fall off more recently. And eventually I'll correlate these more specifically so you can compare them side by side. And then finally, the last survey is the ventless trap survey, which is most similar to the methods used in the fishery. And this is done by covering the skate vent in the parlor of a lobster trap with a finer mesh to hopefully retain more juvenile lobsters than you would otherwise. Here's the catch per unit effort of the ventless trap that pretty similarly follows the trend to the trawl survey. This is also broken into 3 regions, and it's randomly sampled across the state, and the traps are dropped on 4-night soaks. And yes, it's very comparable to the trawl survey catch per unit effort. And here I further broke it down by region, and it also follows that trend of the more northern regions having more volatility, while further south it seemed to be more of a maintenance of the juvenile population that already exists. So yeah, this is an interesting, it offers interesting information what might be happening with settlement in the northern regions, again, as I was referring to before. Here are the correlations of the inshore trawl survey versus the settlement index survey. And there's no statistically significant relationship between the trawl and the settlement survey in terms of juvenile lobsters on the right in that case. And that's interesting because these both contribute to the stock assessment. So it may cause us to have to reconsider how we weigh these surveys in the stock assessment. Versus the ventless trap survey versus the inshore trawl. And these surveys track very well, which gives us confidence in how they contribute to the stock assessment. All right, second part of my research was my own personal study. So I'm kind of segueing out of the data analysis more into the work I did in the field. So the Appledore study that I did was a ventless trap survey using 3 pots that I randomly deployed throughout the month of July around the island in depth between 5 and 15 meters. I tried to stay consistent with the 4-night soaks of the ventless trap survey that the state does, and also tried to stay consistent with mackerel as bait. And every time I went out to haul, I recorded the location of the trap, its depth, the temperature of the water at the time, the salinity. I did an individual ID of whether it was a crab or a lobster. And then I measured the carapace length, the sex of the individual, the egging status, and the cull status, meaning whether it had both claws intact or it didn't. Here are the locations. I dropped the traps around Appledore over the month. And an interesting finding, I guess, that happened as I started to get out in the field was helping Sebastian with his crab work that you'll hear about later. But when we were hauling crab pots, we noticed a lot of lobsters were being caught in them. So it gave me a larger sampling of individuals than I was expecting. However, I didn't include the crab individuals in the aggregate catch per unit effort because the effort wouldn't be standardized. I try to keep all the pots the same, but it's still just interesting data about, how many individuals are out here. Here's the catch per unit effort of the study on Appledore, broken down by pot. You can see the proportion of adults to juveniles in the crab pots, obviously selected better for juveniles just because it has a smaller entrance and is meant to catch crabs. However, the proportion stayed fairly consistent between the three pots, although pot one had significant more catch for unknown reasons, maybe just by chance where I was dropping it, but it was also a yellow pot, so color can be a consideration in that. But that's why I'd like to scale this study larger, so you kind of break down the noise that can occur just with different luck that you have with traps. Next is the comparing the inshore trawl catch per unit effort that I showed you before to this, and the age structure proportion is significantly different between these two things, showing that there are a lot more adult and larger lobsters out around Appledore compared to like at large the rest of region 1 and the rest of the state of Maine. Here's the size distribution of the survey I did around the island, and it's slowly starting to approach that bell curve shape that you want to see with size distributions. So with more data, it would be more accurate, but I was able to record 68 individuals throughout the summer. Yeah. Then these are the length comparisons of the inshore trawl data to the survey around Appledore, and there was the median carapace length was 15 millimeters greater around the island than the rest of region 1. So that just again goes to show how much on average the lobsters were that I caught than the trawl did. Next is just showing how the kind of the breadth of the size distribution. It started a little more narrow back in the day and it's starting to slowly widen. You can see there, and that could possibly signal of an improved recruitment from settlement area expansion like I was talking about, as well as some of these larger lumps in the bottom being strong breeding cohorts, promoting strong juvenile cohorts as the years went on. But this would be something interesting to look into in the future as well. So here are the major takeaways from the research I did. First, the settlement index survey doesn't track too well with the trawl survey and the ventless trap survey. And this is something to keep in mind for the next stock assessment on how much or the different fishing methods that are used in these surveys and how that could affect the lobsters they're catching, but also which one you're weighing more than the others to try to estimate the population. The rocky intertidal regions offshore could possibly act as a haven for larger lobsters, as we've seen in this case, as opposed to juvenile lobsters maybe preferring the gravel and the mud habitats of the deeper water to settle on. And warming waters may be causing an expansion of lobster settlement regions for the time being. You've seen that in the regions further north, but on the other hand, this could also be a recession of lobsters out of southern regions as the temperatures are exponentially increasing. And future work that could be done is, scaling the size of this survey across different depth strata and just making it larger for a larger sample size. Investigating substrate preference, like I was just kind of referencing, could be very interesting to see, like specifically maybe with cameras, where lobsters are preferring to settle, as well as the camera monitoring and the traps. I was able to experiment a little bit with this summer. I have a cool video if anyone wants to check that out later of what's going on in the trap. But the camera monitoring could also be used to optimize the soak length for fishermen, and so they could apply the same effort and hopefully have more landings or apply less effort with the same landings. And then finally, I want to acknowledge everyone that really helped out with this project this summer. Importantly, my advisors here, Mike and Rebecca, thank you so much for advising me in this project, as well as Dr. Celia Chen at Dartmouth, Dr. David Buck for helping get us out here, and Kathleen Reardon at the Division of Marine Resources that sent me all this survey data, Bill Kneisel for funding the project, and the Shoals Marine Lab staff. Like, nothing could happen out here with everyone that works so hard, and there's a lot of really selfless people that have we've met this summer and have got to know pretty well. So been grateful for that. And then last but not least, all the surges. It's been great to get to know you guys and do work with you. So yeah, really, it's been a privilege to be out here this summer, and it's been pretty inspiring. So that's my project. I went a little long, so I probably have to keep the questions brief, but
Jay, you can unmute.
definitely. I was able to meet Reagan and Larson Foy early in the summer, and they're the family that like the main lobster fishermen around the Isla Shoals, and was able to be in contact with Reagan specifically over the summer with any questions in terms of the egg ratio that they were catching throughout the summer, as well as just different considerations that they had around the project. So it was exciting to be able to work with people that are lobstering around here and see their opinions on some of what's occurring. And they were interested by the juvenile distribution I found around here compared to other areas, because this time of year they start to move their traps in the deeper water as it's so warm around the island. So this research could be helpful for them specifically.
And Owen Nichols says, great job, Kieran, way to represent Brewster and Dawson.
Any other questions? Maybe one more. Yes. Yeah, that's a good question. On average, I'm not. Oh, yes, repeat the question. So this is kind of about the mobility of lobsters and whether they have like territory that they would prefer to stay in. And read a few papers on marker capture of lobsters that showed a large range of how far they can move, all the way from like down south of the Cape around Nantucket, that they've seen some come up to the Gulf of Maine. But yeah, on average, there could be, like, I'm not exactly sure about that. Some lobsters may spend their lives in a more localized area. Yeah. I get some help in this project. Thank you so much. Presentation.
So. Yours is. Is that Kieran's video or yours?
Yeah, sweet. So video's coming soon. Thank you so much to everyone for coming out. Really excited to be here right now. And my name's Sebastian, and I worked on the competition between the Jonah crab, Cancer borealis, and the green crab, Carcinus mayanas, in the southern Gulf of Maine. Just to outline things quickly, first I'm going to introduce the interguild species, discuss warming in the Gulf of Maine a little, define my own methodology, explore my results, and then talk about the biological implications of this study. So let's talk about the Jonah crab. Jonah crab for a long time was a bycatch for a lot of lobstermen. But as landing slightly decreased, as Kieran was talking about, they emerged as a fishery. And up until 2015, there was no management on them. And to this day, there's still no stock assessment. So difficult to really know how many Jonah crabs there are out there. But they're known for their non-aggressive behavior. They range anywhere from Newfoundland all the way down to Florida. So they have a expansive range, can hang out anywhere from the shallow intertidal all the way to 800 meters of depth. And usually we find them in about 6 degrees Celsius to 14 degrees Celsius. And what's really interesting that I got to observe a lot this summer is that a lot of the females will come in these summer months and early spring months for what is speculated to be mating and molting reasons. So we're seeing a lot of them right now. In terms of research, like I said, it's an emerging fishery, so there's not that much research done, but currently we're trying to understand more their growth as well as their movements. So that's a lot of tracking data. And then a really interesting study at UNH that's going on is about their survivability after claw removal, because that's another option for the fishery. Just remove the claws, throw them back. So the famous European green crab that Madeline was talking about, the USDA and the US Fish and Wildlife knows them as one of the most wanted invasive species. And they came from Europe in ballast water in the 1800s. As you can see, they're not always green. We got some varied coloration, but they're swimming omnivores and extremely, extremely aggressive predators. And they've had severe consequences in the Gulf of Maine, especially in recent years. As the waters have warmed a little, the winters have been a little more forgiving to them. And so their populations can survive and strive, really expanding their range. Finally, another member of this crab guild is the Atlantic rock crab. Very similar range to their cousin, the Jonah crab, but these guys tend to come into the shallower waters in autumn, traditionally. So to talk a little bit about tidal regions, they can be broken down into 2 main regions, the intertidal and the subtidal. So we have the lowest low tide line, and that delineates when the tide comes as low as possible, that delineates these 2 zones. And like Madeline was saying, the subtidal is not a super explored area. And so that's where I decided to base my focus and where we see interaction of these green crab, these Jonah crabs, and these Atlantic rock crabs. So I wanted to understand how they interact together. That took me to the following question. How does climate change affect the competition between the Jonah crab and the green crab in the subtidal region of the southern Gulf of Maine? Really important thing to establish is there's a sizing difference here. So of course, the green crab, a very aggressive predator. However, the Jonah crab is a little bigger. And so I was kind of curious, can this size difference and this claw difference overcome this aggressive behavior? What do these interactions look like? To understand the sizing, it took a lot of trapping throughout the island, different spots, and try and get the distribution and break down that distribution. As Kieran discussed, there's significant warming in the Gulf of Maine. It's warming faster than 99% of our world's oceans. And we see a lot of global aquatic heat waves going on, which is when we see a significant warming period. And it's a known effect. It's a known fact that there's a metabolic effect on crabs from this warming. So as we can see, with increase in pH and temperature, we see a decrease in the metabolic rate, as well as an increase in the thermal tolerance. So these crabs tend to spend a lot of their energy adapting to the temperature they're living at, rather than spending their energy hunting and reproducing. So talk a little bit about my method. I trapped all over the island, similar to Kieran, worked with Kieran. And these are some of my top trapping spots. And this was done to collect crabs for the lab and also to get an understanding of how size affected the distribution of these different species. Where are the big guys? Where are the small guys? do they prefer exposed or protected areas of the island? So here I am collecting the trap. And this is a really cool trap right here because it really shows the inner guild nature, the overlapping of these predators. You see there's some rock crab in there, some Jonah crab, some small green crabs kind of hidden under the vent. And then we also see the American lobster, which is a key predator here too. And here I am measuring the size of the crabs, just get an idea of who's where. And then after that, I would take them into the lab. In the lab, to minimize interaction between different species, I separated all the crabs by species in these sea tables, as you can see. When they were there, then some crabs were acclimated to ambient temperature of Appledore Island, whereas others were exposed to warmer temperatures of 4 degrees Celsius warmer than the ambient temperature of the water that was being pumped from Appledore Island. And then once again, before any trials began, I also took measurements on the carapace of the crab, the area of their claws, as well as their sex. Then here, before any trials began, as you can see there, the trials occurred in an aquarium. There was separation of the predators, but they had a 30-minute acclimation period to sense each other's presence, as well as sense the presence of a common prey, which in this case was the mussel. And then trials, these feeding trials would happen for 15 minutes, in which I would note the first species to feed and the timing of this initial feeding, the amount of aggressive altercations, and what crab was engaging those and the timing of those, as well as the time, the percent of the time that each species controlled the prey for. So let's look at some results. So here, looking at these graphs, this one on the left right here shows along the y-axis time that the Jonah crab fed for, and along the x-axis we see the size of the green crab. And this is all specific to large Jonah crabs in this instance. And so we see that the large Jonah crab is able to compete with small and medium-sized green crabs. However, when it comes to large green crabs, they're completely outcompeted, as you can see. And also this graph nicely exhibits the behavior that we see in larger Jonah crabs, just because you see there's very few attacks occurring. And that's just because they tend to back off from any predators and minimize interactions. So when we look at medium-sized Jonah crabs, we see a similar trend. They're able to outcompete the small green crabs, but not larger green crabs. And we also see, here's an interesting observation, that when medium-sized Jonah crabs attempted to engage and compete with larger green crabs, they were completely outmatched. And this increase in aggressive behavior did not result in more feeding success, as you can see there. Finally, looking at the small Jonah crab, we saw that they were actually able to compete with the smaller green crabs, which is following the similar trend that as the size of the green crab decreased, the competition of the Jonah crab increased. And we see that the small Jonah crab exhibits more aggressive behavior and engages in more competition than the larger Jonah crabs. And so what happened with the warming? So if we look here, this is specifically honing in on the large Jonah crab and its interactions with small and medium-sized green crabs. So as we can see here, that in terms of the amount of time that the large Jonah crab fed for, with small green crabs, for example, they fed more than 75% of the time. But as they were acclimated to warmer waters, we saw that these same crabs were feeding less than 50% of the time on average. And we also see that this warmer water resulted in an increased aggression of these Jonah crabs. However, this aggression did not result in more successful feeding, which meant that more energy spent and less feeding, less success overall. And then when we look at the small Jonah crabs and their interactions with specifically small green crabs, we see a similar trend that their overall feeding time plummeted with the warming of the water, a mean time of 45% that decreased all the way to a mean of 31%. But we do see a minimal fluctuation in aggressive behavior with this warm water. So the small Jonah crabs were not changing their behavior as a function of this warm water. So to talk about some biological implications. So as I was mentioning, with this expansion of the green crab into a more northern range here in Maine, as well as expanding their range into a little bit deeper waters, especially as other predators tend to move into deeper waters. This is really concerning for the population of Jonah crabs, especially these females that come into the shallow waters. We see that they will and can be outcompeted by green crabs. And furthermore, we see another observation is that potentially at different life stages of the Jonah crab, they will adapt their behavior with more aggressive behavior from the smaller Jonah crabs who don't have the same power of their claws. And another not as featured part of my study that working with the Atlantic rock crab, they're in competition with the green crab. There's no real ability of the green crab to outcompete the rock crab. So it's more the Jonah crab that is threatened. Yeah, and I would like to acknowledge the following awesome people that made this possible. Thank you to my mentor, Celia Chen. Thank you to Mike and Rebecca. Thank you to Dave Buck. Thank you to everyone at Shoals. Thank you, Dartmouth. Thank you, Bill Kneisel, for the funding. Thank you to all of the Shoals Marine Lab staff. You guys are all really awesome, and I've learned a lot from you guys. And thank you so much to my fellow surges. Really helped me be here today, and appreciate you guys all. You guys are all awesome people. Yeah.
So I have a question about, again, what are the different sizes of, when you say small, medium, large? Is a large green crab equivalent to a medium Jonah crab?
No, just because of the nature of Jonah crabs being so much bigger than the green crab, that's not a realistic like interaction, I guess. And so that was kind of based on what I was trapping. And I would find all sorts of different size green crabs interacting with all sorts of different size Jonah crabs. So the small, for example, small green crabs are smaller than the small Jonah crabs. They're not the same size. It's kind of like an adjusted ratio. Yes. Yeah, so I would say this is really like a first step study. And other behavioral studies have been done at Appledore, in which like a cage was dropped into the actual subtidal region. So this is just preliminary, but yeah, those factors are definitely real. And there's also a possibility that the Jonah crab is, less used to human interaction. And so they're more affected by this. So yeah, then the next step would definitely be taking this into the water. Yeah. Sweet.
The first part is how long the crabs were acclimated to the warmer temperature?
Yeah, so kind of just by the nature of needing to get things rolling, the shortest time that some were acclimated for was 2 days, which I recognize is it's really difficult to do temperature-based experiments just because crabs will have years to acclimate to warmer water. But some were acclimated for up to 2 weeks, 3 weeks.
And I'm also curious, why do you think that the crabs were feeding less but more aggressive at lower temperatures, and you see that in most species.
Yeah, definitely saw. Oh yeah, repeat the question. So the question is, if I saw more aggressive behavior with both the Jonah crab and the green crabs, and why I think they were feeding more. So I think part of that might be the nature of the warm water increasing metabolic needs. And so the reaction could be to be hungrier and to have more aggressive behavior. And that could connect also with that short acclimation period. That like initial exposure to the warm water caused that sudden aggression. Yeah.
What is the definition of aggression?
Yeah, that's a great question. Thank you so much. The question is about like what an attack looks like, what an aggressive behavior. So yeah, one metric for that is like I guess an attack. One crab trying to take the prey away from another crab. Another one would be how quickly they approach the prey and the amount of time that they spend with the prey. But mainly, yeah, it's that attack towards the other crab and desire to take over the bait.
And there's some baseline only where how long it takes, but there's no other crab. How long it takes to now get out there.
Yeah.
Mhm,
Yeah. Yeah. So in that case, yeah, before I started my trials, I ran some initial trials to see what that looks like. Not featured here, but they showed that yeah, the Jonah crabs would approach the prey and quicker than they would in the presence of green crabs. Thank you so much.
Okay, we have a quick break to grab a coffee.
Can we try to kind of fast forward the coffee break and be back here in 11:15, I think is what we'll be right back to track.
Great job.
Oh, wow. Hey, good job.
I know.
They're all really how it comes together.
Just, this is what you're looking at. This is recording. We're still recording. So you just see this. And then all you need to do is just hit the space bar.
Great, right.
So it should just, should keep going. Yeah.
Huh? What'd you say? No, we are smiling. Yeah, we are.
Right now, we're reviewing Casey's screen, but then, okay, when we come back, I'll screen share for you. Yeah.
It's like such a cool yeah, me too. It's like it's great. I think I ran here. Here. That's true. Yeah, so I think that's really sad. Yeah. Yeah. Yeah, it was also a lot of sense. Last year specifically. So, yeah, that's kind of. So Democrats really brings down to the lobsters. So that's kind of a
Well, that should be good. I wish I could find a way to get stuff on the top, but most of my stuff is below anyway.
But I think actually
Everything's great. You can also, if you're curious, I think I can hit the reset button and then you can like keep track of your time.
I don't mind.
Y'all have it.
Yeah, I know.
Yeah, I gave up. I was like, I'm not gonna keep track of
I'm
your
not
time.
track
Any
on
other
it. Whatever
questions?
happens, happens.
Yeah, seems like y'all are keeping pretty good pace.
Yeah, we caught right back up. You got it.
Yeah,
You want
Okay.
me to yell at people or
you yell at people.
I saw some you need to take. Thank you so much.
All right. Hi, everyone. Thank you all for sticking after the break. My name is Bella, and I'm one of the marine mammal surgeons this year, along with Sarah, who you'll hear from next. But my independent research project looked at the effects of temperature on molting phenology of harbor seals out here at the Isles of Shoals. So first things first, I'd like to orient you all towards the topic of what is molting. So molting is a process that all seals go through, but it looks different depending on the different seal species. So luckily for me, the two species that we see out here, the gray seal and the harbor seal, molt in a very similar fashion. So what they do is they undergo a month-long process in which they shed all of their old fur, usually starting in the center of their body and then ending at their heads and their limbs, and they grow back new fur underneath. They do this process right after their pupping and breeding seasons. So for the harbor seals, which are the pups that you see up here, they're pupping in the early summer, so they are molting in the mid to late summer, and then the gray seals are pupping in winter time, so they are molting in the spring and early summer. So here's what their annual cycle might look like. So they've got their pupping, and then they're taking care of their pups for a few weeks, and then they move on to mating, and once they're done mating, they will move on to molting for about a month, and then they'll continue on with their lives until they come back for pupping. So molting is a very energetically costly process, and it also puts the seals in a vulnerable position. So you might wonder why they bother going through this process at all. and they have two very good reasons. The first one is getting rid of their longer hair, which can create drag for them in the water and make them swim slower. But the second is that their fur also provides a form of natural UV protection. So when they're shedding off all of their old and damaged fur and growing out new one, it's like they're putting on a layer of sunscreen and a cute little sun hat. So now that we know what molting is and why it's important, I'd like to talk about a few constraints that the seals have to their molting. So as some of you might know, seals spend about half of their time in the water and half of their time out on land. And when they're in the water, they're mostly foraging, but they can also sleep in the water, and they'll also flee into the water if they feel like they're threatened on land because they're a lot more agile in the water. The downside to them being in the water is that they lose heat at a much quicker rate than when they're out on land. So to combat this rapid heat loss, they perform a process known as peripheral vasoconstriction, in which they limit blood flow to the blood vessels in the periphery of their body in order to promote blood flow in their core and their brain to protect their vital organs and maintain those healthy. However, when a seal is molting, their blood flow is needed elsewhere. They need to send their blood out to the skin in order to promote hair growth and to molt efficiently. This means that they aren't able to go into the water as much, and so they're hauled out for a longer period. And like I mentioned before, they're foraging when they're in the water. So when they're molting, they're essentially going through a month-long period of fasting. Luckily for them, their bodies are completely adapted to this, and they have hormones that regulate their metabolism and lower their energy consumption. So they are able to survive this period with low amounts of food. These hormones are regulated by environmental cues such as light and temperature, which prompted my research question. Has the increase in temperature in the Gulf of Maine affected seals' molting phenology? And I hypothesized that the change in temperature would shift the molting season earlier each year. So not only is this an important topic, is this an interesting topic to me, but studying phenology, especially in the Gulf of Maine, is also a great indicator of the impact that climate change might have on an ecosystem. So in a paper written by Staudinger et al., they conclude that changes in phenology, which is the seasonal timing of recurring events, have emerged as a primary indicator of species responses to climate change. So this article focuses mostly on changes in phenology in seabirds, plankton, fish, and macroinvertebrates, but they don't touch on marine mammals at all. However, since seals are a sentinel species, which means that they provide an early warning that ecosystems are out of balance before other species might display those effects, I figured that studying their phenology might lead to important conclusions. So in order to address my questions, I needed to do a few things. First, I needed to collect the data. So for everyone in the room, if you're new to the island, we are standing and sitting where the pretty pink smiley face is. But Sarah and I, we go out to Duck Island, which is right over there, and we conduct, and it's circled in red up on the screen, and we conduct seal surveys out on a boat, and we're looking for entanglement, seal populations, injuries, and anything else out of the ordinary. So this is Duck Island, and previous surgeons have been doing this for 12 years, so we are adding to a historical data set now. So each year, roughly 20 to 25 surveys are conducted, and we go out on beautiful sunny days like this one, but we also go out on foggy and rainy days, with our only constraint being that we go out at low tide, so we're able to see the highest amount of seals. So once we've taken all of our images, we come back to the lab and we look at them and we upload them, and we're able to create graphs such as the one you see here. So on the x-axis, we have survey dates ranging from 2011, which is when the project started, to 2023, which is right now. And then we're looking at the number of harbor seals found in each survey date. And as you look at this graph, you might notice that there is a great variability in the number of seals that are found on each survey, ranging from over 600 to essentially 0. And because of this great variety, I realized that my study had to look at proportion of molting seals and not straight up numbers in order to correct for the variability in the number of total seals each survey. So once I knew what I was collecting, I had to make sure that the data that I was collecting was correct and accurate. So I did have to be a little bit picky with image quality, because I wanted to make sure that I was accurately reporting a lack of molting because there was no one molting, and not because I was unable to see the molting or tell that they were molting. So as you can see in this image, there are seals there, but you really can't say much about them besides the fact that there are seals there. So I did have to discount certain images because of fog or glare or distance or poor image quality, whatever inhibited me from properly seeing the seals. So once I knew which images to use, my next step was to figure out what molting actually looks like. So these are all examples of molting, and as you can see, they look very different from each other. So a large part of my early process was sending out images such as these to my mentors and to other individuals who are a lot more seasoned with spotting molting out in the field. And I continued to send out these images until I was confident in my ability to accurately find molting. So in my journey, I encountered a few issues with looking at gray seal molting specifically. The first is that they tend to haul out in locations that are lower to the water, which means that they are wet. So it's harder for me to differentiate between a molting seal and a seal that's been splashed. In addition to this, they hang out in crowds, so I'm unable to see the majority of their bodies to see if they're molting. And finally, since we only start collecting data in June, we're only able to see the tail end of their season. So as a result, I was only able to confidently find 2 molting gray seals. So my project became more focused on the harbor seal molting. So that's what I continued with. And molting data wasn't something that has been collected on the seal population out here at Duck. So that meant that I had to personally go through images from the past 12 years, which meant over 200 surveys, over 9,500 images, and I just looked for any and all molting harbor seals. And these were the results that I found. So I'll orient you as to what we're looking at. So on the x-axis, we have the survey date, and on the y-axis, we're looking at the percentage of harbor seals molting on that date. And then each box represents a year. As you'll see, 2014, 2020, and 2021 aren't included, and that's simply because we didn't have good data for that. I also included a minimum sample size of 30 seals for each survey in order to avoid skew in my data from having small sample sizes. So these are the results that I got, and as you can see, there's a little bit of noise, but you can start to see a trend. So as I mentioned earlier, the timing of harbor seal molting tends to be in the mid to late summer in general, but like the regional differences, there are regional differences. So it looks like here at Shoals, the peak of the molt extends sometime in August. Unfortunately, we're unable to say exactly when in August, since the August data that we collect tends to be from the first week and a half. So we're unable to see if these upward trends continue on or if they've reached their peak. And this will become very relevant in a few slides. So next, I wanted to introduce the variable of temperature. As multiple other surges have pointed out, the Gulf of Maine is warming at an increasingly alarming rate. And so here you can see the summer sea surface temperature for the Gulf of Maine spanning back about 40 years. But the years in which our surveys were conducted are in the blue line, the blue dashed line. And as you can see, the trend is upwards. So in order to compare my data to the temperature, I needed to bin it into months. And again, keep in mind that the month of August only has data from the first week and a half. So here again, we're looking at percentage of harbor seals molting, but this time we're comparing it to the average monthly sea temperature. So when we plot it and we run a linear regression, we can see that there is a significant correlation between temperature and molting. And if we look at the month dots, we can see that the June temperatures tend to be lower, and the July and August temperatures are in the higher range and also are experiencing a higher amount of seals molting, which is consistent with the literature. So now that I determined that there was a correlation with temperature, I wanted to see if temperature affected the phenology, which again is the seasonal timing of recurring events of the molt. So I compared the percentage of molting with year and month. Again, years on the x-axis, month are in colors, and then we're looking at the percentage. And I wanted to see if there was a yearly effect on which month the seals are molting most in. So these were my results, and I ran an ANCOVA and I got a p-value with less than 0.001, with the most significant effect coming from the comparisons of the month of August. So it's clear that there is a significant change in the amount of harbor seals molting in August each year, but what does this mean for my hypothesis? So at a first glance, I thought that this meant that the molting season had been pushed back later into the season, but keeping in mind that the data from August is only taken from the first week and a half, I later concluded that it's more likely that the molting period had been pushed earlier into the season. So in 2012 and 2013 and at the beginning, you can see that it looks like there's a very low percentage of seals molting in August, which is likely because sampling in the first week, which again is where the August data is from, excluded the peak molting observations, which in the earlier years were likely to be after our sampling period and later in August. Comparatively, this year, it looks like there's a lot more molt in August, and we haven't sampled any later into the month. So this leads us to believe that the peak molting week is moving earlier and earlier into our schedules, and into recent years, it has moved if not into our sampling schedule. It has moved closer, if not into our sampling schedule, which is why we're seeing an increase in August molting. So some key takeaways from my talk that I'd like to leave you with. First is that harbor seal molting in the Isles of Shoals likely extends into August, but to know the exact peak, we need to do longer survey periods. It's also possible that early effects of climate warming on seal molting are possibly visible and might be shifting molting earlier into the summer, but really further research needs to be done in order to confirm and expand on these findings. So some future directions that I would love to see this go into are to compare the molting of both gray seals and harbor seals throughout the Northwest Atlantic. It'd also be really awesome to send out crews in the spring out here to Shoals in order to gather data on the gray seals, and also to extend the stay of the surges or have other people come out here to stay beyond August in order to capture the entirety of the harbor seal molting. However, I think the most important study would be to determine what, if any, are the health implications for seals to be molting later in the season, and how is this shift affecting them and their role in the ecosystem. So I'd like to acknowledge that my project would not be what it is today without all of the incredible people up on the screen. Thank you to my on-island and off-island mentors, my fabulous SEAL partner, Sarah, Noah for stepping in when Sarah had a concussion, the incredible captains who took us out on all of the boat surveys, and we'd have no data if it weren't for them. And of course, everyone here at SML who is just keeping us fed and going and alive. And of course, all of my fellow surges, much love. Yes. Thank you. Yeah, so you're asking if the molting earlier is cutting into their breeding season at all. So we see the like new pups earlier on in summer, and the molting doesn't look like it's been moving that much further back. So I would say no, it doesn't look like it's impacting the breeding season. Yeah. Yes.
Yeah, great talk. You obviously have very clear correlations with temperature. And I think we just kind of outside there, we're thinking about what else might be correlated. Like thinking about, well, if it's strongly correlated with temperature, what else might be correlated with temperature?
Oh, sure. Yeah, I mean, I think a big thing is that they are molting mostly in August, so there is an increase in temperature in August, like throughout the summer. So it's possible that the temperature, the temperature only graph was capturing only the August data. But when I looked at it, the August and the July temperatures seem to be very, very similar. And so I no longer think that that's the case. I think that could just be a correlation. Did that answer your question?
A little bit, yeah. Just thinking about other factors that might also be correlated with temperature. So you can imagine things like maybe disease is important.
Oh, yeah. Yeah, of course. Like for seals in general, disease definitely could be correlated with temperature. Another thing to think about is their diet. So with the change in temperature, the fish are moving in different ways. And so if their feeding period isn't matching up with when the fish are there, that can also impact their health. Yeah, it's great to think of it at a very ecosystem level, because the temperature can affect all sorts of moving parts, which can then affect the seals. Yes.
Fantastic. Can you foresee the use of technologies like UAVs or artificial intelligence to help with image analysis and helping collect better data or more data?
Yeah, absolutely. I was a little bit limited, especially in the earlier years. We had a lower quality camera. So yeah, in implementing technology and AI to look at not only molting, but also getting better pictures of the molt is definitely something that I think would be great to see in the future. Yeah. Yes.
Hello. My question, it's more of a personal question. I would like to know what your favorite part of this whole research experience was for you.
Yeah, so this is my brother actually. Um, yeah, I think my favorite part is just being out on the island and being able to meet so many incredible people, and also having all the opportunities that these incredible people have allowed me to go out on. It's also been really cool to watch the birds grow up. I don't know if anyone who just got on island, if you've seen the little the grayish gold chicks, they were menaces at the beginning. They were running around and chirping and just being absolute craziness. But that's been a big favorite of mine. Yeah. Spacebar?
There you go.
Oh, wow. Okay, cool. Hi, guys. My name is Sarah Morris, or Spirit for some of you guys. And I am a mechanical engineering major currently at Arizona State University. I wanted to briefly mention that as a mechanical engineering major who loves the ocean, there isn't enough said about how important it is to join biology and engineering together. There's a lot to be said about the environment and how we as engineers can take inspiration to further our tech. And I'm very passionate about biomimicry specifically, and I would love to see inventions in our world that is taken from the world like around us. So that's kind of like a bit of context for why I, as a mechanical engineering major, am here at a marine biology lab. So yeah, without further ado, here is my presentation on seal vibrissae and why they are superior to all kinds of whiskers. So just to kind of go into what the word vibrissae actually means. They're whiskers. They're a fancy term for whiskers. They are tactile, stiff hairs that can help the individual process their environments. Most mammals have vibrissae. You guys see vibrissae a lot, especially if you have any pets like dogs or cats. These dogs and cats and other animals that or pets that I will be presenting to you guys are from the sturges and from Cormac the chef. That's the dog picture all the way on the left. That's Aggie and her really amazing vibrissae. This is a really good photo. So yeah, all these animals have vibrissae, and this is very important to note. So the similarities between all these animals, dogs, cats, and hamsters, they have smooth and symmetric whiskers on all sides. This is kind of like a basic drawing that I drew about what that looks like if you were to look at the whiskers from all sides. You pick up a whisker from a cat, dog, or a rodent, and from any side, it's smooth on all sides. So this is really important to note, especially in context of seals, because seals do not have that at all. They're very asymmetrically shaped whiskers. Their morphology is very different, even between between like the species. So like, as you can maybe see, like the gray seal, the harbor seal, the harp seal, the whiskers that I'll be going into today are more wavy, but like the sea lion and the bearded seal, they do not have any waves on their vibrissae, which is very interesting to note. So yeah, we'll get into that. Here's a harbor seal picture in kind of a zoomed in photo of what those wavy sides look like. So why does shape matter? Studies show that when you give a seal some headphones and you blindfold them, they can maneuver through the water perfectly. So here's a seal, and he does not have his ears or his eyes. So they put a ball in the water, right? And they say, go fetch. And he got the ball perfectly, as if like he didn't have any of those constrictions on him. So here's a more in-depth photo of what that looks like in context to like prey or like any species of fish in front of him. He will like make movements in the water. Those movements will transfer to the whiskers and the seal will process them. Whiskers will move forward, and he will then know not only that there's somebody in front of him, but he'll know exactly what type of species of fish it is, if it's a fish, if it's a shark, if it like all kinds of information that he needs, which is something that's very not normal for other kinds of animals like cats, dogs, rats. Like you guys as pets can't do that. So this is very important for foraging and for watching out for predators. So here is another illustration that I've made, kind of going in depth of what it would look like, what these like different sides of these asymmetric whiskers look like. This is kind of a drawing of one whisker if you viewed it on different sides. There's like two sides, two flat sides and two long sides. Kind of like a rectangular prism of sorts. Here are some other illustrations that I've made of the different seal species that I'm going to be covering here today. On the left is a gray seal, harbor seal, and a harp seal all the way on the right. As you can see, they have similar patterns to their undulating whiskers, which has me wondering exactly like how these whiskers interact with their environment on a mechanical level, which leads me to my hypothesis, which is when you introduce a sound source to a whisker, either to its undulating side or wavy side versus its smooth side, the wavy side will move or process those vibrations more visually than a flat or smooth side of the whisker. This is an illustration of like the same whisker, just presented different sides. So here is an illustration that I made of what like on a more like zoomed in photo of like a harp seal photo. Here are some pictures of it underneath of like the real deal. I'll take measurements from 1 centimeter in, 3 centimeters in, and 6 centimeters in. As you can kind of see in the bottom photos over here that the whisker like waves will be a little bit thicker towards like the middle or the 3 centimeter or 4 centimeter mark. And then it kind of thins out towards the 6 or 7 centimeter mark for this specific whisker. So kind of going deeper into that with the harbor versus gray seal whiskers. Here are 2 whiskers. The bottom one is a harbor seal and the top one is a gray seal. And they kind of look the same. They kind of start out very similar, where 1 centimeter in, 3 centimeters in, and 6 centimeters in. This is very interesting to note for me personally, because the gray seal whisker, if you can kind of see at the top, it's very hard to see. It's very, very thin at the towards the tip, and it kind of thins out faster than the harbor seal whisker at the bottom. I made sure when I was comparing these whisker sizes that they were like around the same length. So both of these whiskers were around 6 centimeters in length. So it's kind of interesting to see that same age seals are both like weanlings or like juvenile seals, and they both had the same whisker length, and they were both like very thin. Where the top one was thinner than the bottom one. So this is kind of a graph that kind of takes all the whiskers that I've sampled from the gray seal, which is the red line, the green line, which is the harp seal, and the blue line is the harbor seal. And it kind of just takes all of the measurements I took from the 1 centimeter line, 3 centimeter line, and 6 centimeter line from root to tip, and observe the variations with undulations or the wavy side and respect to the length. So methodology. I wanted to preface this by saying that processing movements on a scale like this and frequency and underwater acoustics would mean that like I would need multimeters and frequency readings, and I would like to use SolidWorks, like 3D software, and just like technology that you find in like a more professional kind of like lab to kind of analyze stuff that's really, really small and kind of hard to see with the naked eye. But what I got was PK Lab, and I love, and they do not have the type of tech that would require me to look at something that's so small, which makes a lot of sense though, because it's this type of like whisker research is very small and very minute, and the equipment that I would need is very like specific, which led me to kind of brainstorm ideas on how I could measure the whisker movements on a smaller scale, kind of like using that engineering problem solving skill set, which I really enjoyed that side as well. So here's a whisker. Here's how I did it in the first place. There is a whisker, same whisker, flat side, stick it into clay, facing the sound source. This is what it would look like if it was facing the sound source, which if you guys were the sound source, this is how the whisker would look like if it was wavy, and this is how it would look like if it was flat facing you. Here is a video. It shows. So as you can see, it is moving a little bit. So I kind of had to look really close up to the glass to kind of see, is it moving, is it not moving? And the stir in the bottom of the tank is how kind of like the constant movements that would produce those vibrations that I would later like see kind of reflect onto the whisker itself. Okay. And so in terms of distances, I would put the whisker all the way far and test to see if it would move, then intermediate distance, and then close distance. So around like 5, 3, 1 intervals of like distances. And here's another angle of that from the side. So you see the stir is moving, the purple indicates the vibrations, and then the whisker is then either moving side to side from the side angle, and then here is it from the front. So it's kind of like moving side to side like that. And here is my results. So the graph represents the movements of the whiskers. So like how big or small the graph is represents like its strength, stronger in movement or lesser in movement. The right side is the undulating movement or the wavy side when it's facing the sound source, and the left side is the flat side of the whiskers when it's facing the sound source. I want to direct your attention to the top row. This is where the whiskers show the most movement when put to the closest distance of around 1 inch. As the whisker gets further away, the strength of movement decreases as seen in the intermediate distance. And then when the whisker is far away from the sound source, it rarely moves, as you can see on the bottom. Rather than looking at the parts of the graph that show the most movement, like many people will like look at more, what you should focus on are the parts of the graph where you see little or no movement, which is towards the bottom. This is kind of like the best way of seeing that, like when the flat side is facing the sound source, it's not really moving as much or as strong as the undulating side, because the undulating side is more sensitive based on these results, which, you know, the undulating side of the whisker is more sensitive to vibrations than the flat side. Here's a seal. You see how, yeah, that's its vibrissae closer up. But yeah, so for future plans, I would like to learn more about the, do more in-depth analysis of the whiskers itself, the morphology, the shapes, and kind of learn more about how I can integrate that, or how we engineers can integrate that into ocean sonar or just ocean technology in general. I would love to dive deeper into the seal anatomy, observe seal vibrissae and how the brain decodes movement into information on a biological level, then convert that into a mechanical process. That's like a very elaborate later step process. Kind of like complementing biology with engineering, because I think that those are very important to kind of not only complement the seals, but also complement us as like since they're better in their environment, they live in the ocean, we should learn from them in order to learn more about our oceans, in a sense. Like they're kind of like the bridge to get to that area. And yeah, I think it's also interesting to note that since the seal vibrissae is different from each other, and since, like, for instance, bearded seals, they do not have waves in their vibrissae, wonder why that is. There's also like harbor and gray seals, they're very opportunistic with their prey. But like species like the harp seal or the bearded seal or the walruses, they're very specific in their prey. So even though like the harp seal is very similar looking to the gray and harbor seal whiskers, like the ones that you see on top, their prey is different. So it's very interesting to see why that is. And I think that it would be very interesting to kind of dissect that and to look more into why that is. So yeah, references, and I'd like to thank my mentors, Dr. Dre, Lisa Sett, Center of Coastal Studies, for sending me the whiskers, because that's a very difficult process. Thanks to Bella for being just an amazing SEAL partner in all things. Thanks to my PR team, Wolfgar, for helping with presentation and all kinds of science communications. Thanks you to the captains, Roger, Steve, and Heather, for taking us to Duck Island and for all the amazing stories that you guys have to share. Shoals staff, kitchen staff, island engineers, everybody in between, you guys are amazing. And last but not least, I'd like to thank my surge interns. You guys are the best. I love you guys so much. All right, you guys have any questions? Yes. Good question. So the question was, why is the undulating side of the whisker more sensitive than the flat side? And then what would happen if there were different frequencies introduced? So in other studies that I have read, I think personally that the undulating side seems to have a larger surface area. That's like, I guess, like a more basic type of like thinking about it. Like the more surface area you have, the more like the more vibrations will have like a surface to like touch. So that was like my first thought about why that would be the case. I think there has been studies shown that like the way that the waves are designed or engineered on that seal is kind of blocks out the noise, the like other types of vibrations that are not needed or not necessary in IDing a fish in front of them, but then also inviting the vibrations that are needed to identify it. So like it filters through the vibrations, if that makes sense. I do not know exactly like the mathematics or the exact like mechanics as to why that is. That's something that I'm actually very interested in looking up like outside of here. But yeah, I would believe that that is kind of like the basic explanation of why that could be the case. Yes.
So we saw that the seals were able to do the thing with that sensory deprivation. How often are they gonna be relying on just the vital say?
So seals can dive pretty deep. They can dive up to, or at least the harbor and the gray seals that are found in the Gulf of Maine here, they can dive to 1,500 feet. The waters here are a little bit murky as well, so it's kind of hard to see under the water. So they're pretty, at least in this area, they're pretty reliant on their vibrissae. I say that that is like the most, I'd like to say that's the most important part of the seal, and how they need to kind of go about their life. Sometimes like it gets murky, sometimes it's raining, sometimes there's no sun. So I think that it's pretty frequent that they would need their vibrissae, that they, yeah. Yes. Oh. Yeah, that's a really great question. I've been trying to actually find pictures of seals online that would have a very much very zoomed in photo of that. I don't really see from the research that I've done, I haven't seen anybody talk about that. I haven't seen anybody like get a very clear picture of what that looks like. And I've been waiting for a seal necropsy to come through here. There hasn't been. So in the future, I would like to kind of take a picture really close up of a seal. If any of you guys have access or like work with seals on a daily and want to send me a picture of a zoomed in photo of a seal, that'd be great. Because I'm actually very curious about this as well. Because I think that that's very important in context to this research project in general. So yeah. Yes.
I really appreciated your emphasis on connecting engineering and sciences.
Yeah.
I was curious, would it be worth looking at the modal frequencies of vibrissae and see how they might spectral response to that looks like, and maybe certain frequencies would couple better to them?
Oh yeah, yeah. I think that in general, testing any kind of like how different how different seal species respond to different frequencies, like if for instance, like this is what I'm saying about like for like the gray seal specifically, since the whiskers that I've seen are very thin at the end. I was wondering if like that processed something better in the water, like if that was better for like shark detecting maybe, or if it was like better for like smaller types of vibrations, because maybe it could pick it up better because it's influenced more in the water. I would love to dive into that deeper. I think that that's something that would greatly benefit ocean technology in the future as well, to detect all types of moving things in the water, because we can't obviously see under the water. So that would be really interesting to see how they differ between each species and how we can test that out as well. Yes. Well, I do know that in the past they used to do that. I do know now that there is the Marine Mammal Protection Act that prevents anybody from doing that. Yeah, exactly. I think any kind of experiments that would be like okay to do, I guess, are seals that are already in research facilities or like in captivity already. But I think like the most that they could do is just like blindfold them. But that's as much as I know about for right now. I think the Marine Mammal Protection Act just makes that entirely difficult. Yes. Oh yeah. Seals, sea lions and seals do have similar vibrissae. The thing is though, is that sea lion vibrissae is very like kind of similar to cats and dogs, where they are very like they're symmetric on all sides for the most part. So I do know that while they do relate to like the seals that do have the undulating sides or the wavy sides, it is a little bit more difficult, just a little bit more difficult for them to process the vibrations as well as a harbor or a gray seal would. Hi, everyone. I'm Abby, and my presentation today is going to be on chick diet composition and feeding frequency of black guillemots on Appledore Island. So here's some general information about black guillemots. There are seabirds that nest in rock crevices. They usually lay 1 to 2 eggs. This is their egg, and this is a pipping egg, so you can see a bit crack on the top. They are part of the owlset family, the same as puffin, and the owls of Shoals is the southern border of their range. So this is an interesting location to study their behavior. Here's my research questions. Do different nests have different diet compositions? Do guillemot chicks who experience higher feeding frequency have higher growth rates? And do guillemot chicks who consume fish with high mass have higher growth rates? So these are really diet related, so it's important to know how they forage. So they can dive as deep as 50 meters, but they would prefer shallower inshore water that's under 35 meters. And their mean dive time is 73 seconds, with some dives extending to 150 seconds. So why do we care? Because their diet is a reflection of the state of the ocean, and it's also an indication of fish stock diversity to see if we'll be like overfishing some species or not. This is a video of an adult feeding a sculpin to its chick in its nest. And they're just absolutely adorable to study. So the data needed is the growth rate, how fast do they grow? The feeding frequency, how many meals the chick eat every day, and the average mass of the fish, how heavy. So if you go get a hamburger, you can get a small slider from White Castle, or you can get a Big Mac from McDonald's. It's the same for the birds. You can get a tiny cunner that's roughly around like 30 millimeters, or a huge rock eel that's around 15 centimeters. So these are our study sites. There's 11 occupied nests in Brock Cove, 5 in Transect 24, and 4 in half-tide ledges. So what I do there is I grab the chick. I muted the video because the chick is screaming really badly. So after grabbing the chick, we measure their weight, their culminate length, which is their upper bill length, their wing length, to that would be the measurement of the growth rates. We also bend the birds. So if there's 2 chicks in the same nest, we can tell them apart. We also collect poop and feather sample for further analysis. So I'm going to pass this poop sample around for anyone to look at. So there's poop in there. So here's Noah Milski. He is holding a roughly like 3-day-old chick in his hand. And if you zoom in, you can see their toes are actually colored because those are too young to be banded at first. So we use nail polish to mark the different birds. So gulls, as you can see here everywhere, they regurgitate to feed their chick, and they don't have a fixed location for their feeding events. However, for guillemots, they always bring the whole fish back to their nest and feed their chick only in their nest. So this gives us a great opportunity to study their feeding behavior with a camera. So this is the motion camera, a motion sensing camera we use. We use that to measure the feeding composition and frequency. So I use the number of feeding events that took place in 24 hours on average to measure their feeding frequency. And I also ID the fish they bring back, and I write down the size of the prey too. However, this camera has a little bit like disadvantage because it has a 5-second shot lag. So this would make us a lot of the feeding events that I write down, it will be presumed because I don't really get to see the fish. They're really fast when they're entering the nest. So these are a few examples of our camera setup. The one on the left is the one we have in Brock Cove right now. Well, not right now, but like a few days ago. And that one is not our best setup because although we try different angles, the bird will always enter from the left side of that camera and always catch a lot of gull chicks. And because the burrow is really narrow, so we don't have a lot of choices. And the one on the right is actually a perfect setup in Transect 24 because the bird, if they bring in the fish back, it will land briefly on the rock that's right in front of the camera. So we get to see the fish clearly. So this is an example of a good quality video from Transect 24 too. That's a rock eel, as you can see. And So here comes the fish ID and measurement part. That's a screenshot from the previous video. So I use culminate length as a measurement of the size of the fish. So for this one, it will be roughly 3 culminate length. And the average culminate length of an adult guillemot is around 30 millimeters, 3 centimeters. So these are a few of the common fish we see in the video. This is a rock gunnel. It's a red, flat, long fish. So as you can see in the poop sample, the poop is a little bit red. That's because the major component of the chick's diet is this fish. This is a gull trying to steal a rock gunnel from the guillemot nest. This is a cunner, as you already know from Madeline's speech. We have the smaller version of it because it's like usually this big. This is a sculpin. They're really spiny, and it has like a little bit blue undertone in its belly. This is radiated shanny. It's shaped like a rock gunnel. It's like a little bit fatter and like shorter, and it has a very consistent brown color. However, this is usually what I get to see in the videos because they entered too fast. So even if I screenshot it, adjust the image color, I can't really ID the fish, but it's still very important data for feeding frequency. Here's the results. So we ran through roughly around 10,000 videos with 20 seconds each in total. We have 12 cameras in deployment on Appledore and for roughly 9,000 hours. We seen a total of 1775 feeding events, and we've seen a total of 562 fish. And this is the first time we collect this kind of data with this specific guillemot population on Appledore. As you can see, rock gunnel makes up the most of its diet, which is 268, and then it's the cunner, sculpin, 89 and 85, 43 radiated shanny, and we have Acadia ratfish, bearded rockling, cod, and shrimp, which are all under 5. So I select 3 nests that have good fish data on and made a chi-square test with it. You can see rock gunnel still makes up a huge proportion of it, but for rock oak 2 and heli, sculpin also made up a great proportion, and the cunner for heli 2 as well. So the chi-square test actually turned out to be pretty significant. It means like different nests have very different diet composition. So here's growth rate versus feeding frequency. So these are the individual growth rate curves I generated for each chick we have good data on. So you can see they're really linear. It's age on the x-axis and wing length on the y-axis. So they're really linear and the slope are roughly the same. So I took the individuals, the slope, and pair it up with the feeding frequency I got. So on the x-axis of the next scatter plot, it will be the feeding frequency, the average number of feeding events that take place in each nest in 24 hours. And if there's 2 chicks in the same nest, I will divide that by 2. And the y-axis is the slope. So it looks roughly like this. This actually shows there's some kind of correlation between these 2 factors, and it's negative, which means the more feeding events that happen in 24 hours, the slower the chick grow, which is doesn't sound right. So that's because of guys can see there's like a blue dot on the right corner. That is an outlier. And if you go back to see the growth curve, that one is heli 4A. So it is shorter than the rest, like the other chicks. That's because we only started to collect data on this chick when it's like roughly 25 days old. So the growth curve might slow down like at that point. So if you get that out, you see there's no correlation between these 2 factors, which means there's other factor that's affecting the growth rate of the chicks. So I also calculated the average fish mass they bring back from the length I write down earlier. So that is the formula I use from this literature. And there's a different coefficient for each fish. They don't have this this coefficient for every fish that I'm studying. So I use the ones with similar shape. So this is what it looked like. There's also no correlation. On the x-axis is the average fish mass, and on the y-axis is the growth rate. So, however, if you pair the average fish mass with the feeding rate per 24 hour, there's a very significant correlation between these 2, and the p-value is below 0.01. So it means the average fish, like the higher the average fish mass is, the lower the feeding rate per 24 hour it is. So it actually makes sense because it's like us, you're trying to get goldfish, for example, from 7-Eleven. You probably go there every day because you get like a very small pack. However, if you go to Costco and get like a family size 10-pack, it'll probably be set for the whole month. It's the same for the fish. It's the same for the fish and the bird. So if you get like a tiny cunner, you probably have to go out again really soon to feed the chick again. However, if you get like a huge rock gunnel, the chick might be settled for like 3 hours. So here's some takeaway. Guillemot chicks' diet are more diverse than my mentors thought they would be, even between the nests. So because at first we thought it's only going to be rock gunnels and cunners, however, they turned out to be more. And camera may not be the best tool for feeding frequency because we have really low confidence in the capture efficiency. There are chances that the bird might be entering the nest, but the camera just didn't catch it. And also I mentioned the shot lag at first, so we don't get to see the fish that often. So there might be better cameras we could use. And they're really diverse in fish size. The smallest fish we see is like around 1 culminate, which is like 30 millimeter. And the biggest one is 5.5 culminate. And the higher feeding frequency is correlated with smaller fish. So here's the future direction. So as I mentioned, there might be other factors that's affecting the growth rate. So different species of fish, they have different nutrient component. So that could affect the growth rate of chicks. We collected poop sample somewhere in the audience, maybe right now. Those will be used as DNA analysis for diet that I'll be continuing in fall with my mentor, Gemma Klukas, in the Lab of Ornithology in Cornell. We also collected feather sample for the chicks, which could be used for isotope analysis because the chicks are only fed on the fish that's around this water. I mentioned we can use other types of camera. So from another study that's in 1986, crustaceans like lobster, crabs, and barnacles are found in more than 50% of the adult guillemot stomach. So, but however, we've never seen like the adults bringing back crustaceans to feed their chick. So there might be a significant difference between the adult diet, like what they feed themselves and what they feed to their chick. It's also good because of the food availability in different areas, because that study was conducted in the Hudson Bay in Canada. So here's a fun video of things I found on camera. So I would like to end my presentation with a quote from Lorex. Unless someone like you cares an awful lot, nothing is going to get better. It's not. So acknowledgement. I'd really like to thank Liz Craig and Gemma Klukas, my primary mentors, Mike Sigler and Rebecca for as Surf, my coworker Noah Milski, and Willow. Thanks for she's a current seabird technician on White, and she showed us all the guillemot nests because she's a seabird search intern from last year. U.S. Fish and Wildlife Service, Shoals Marine Lab, Cornell Lab of Ornithology, Maine Coastal Island Wildlife Refuge. I'm going to thank Bella, Maddie, Madeline, Kieran, Sebastian, Sarah, Nia, Morgan, Arena Cormac for coming to grab chicks with us. And Dr. Dave Buck, Flora, Roger, Kate, Ken, Kelly, Merrick, Sarah, Jenna, and all of other SML stuff for supporting. Thank you.
I think really nice talk. I'm curious, like looking at other things as a being matters and stuff around how selective they are. Like is rock metal just the most prevalent fish that's around, or how selective are they being versus what's actually available in the environment?
So this question is about the prevalence of fish around the shoal, Isles of Shoals. So I was trying to do some kind of like chi-square test between like the fish composition in the water and with the fish composition in their diet. It actually doesn't work for me because there's not a lot of overlapping. I tried to see if Madeline have some rock gunnel, and I tried to use the nuisance tow data from Mike because those are different like populations in different depths of water. So I think they don't have a lot of overlap, but it is a very interesting like direction I would want to look at later if we have good data on like what's like in the middle part of the water, like probably. Yes. I actually don't know, but I think it is a very interesting to think about. So like for me personally, I would interpret it as, oh, because my chick is being fed with a big fish, so I can like rest for an hour or something like that. And if it only bring back a smaller fish, because I don't think like they usually decide on like what kind of fish they would get in the water. So that would be something that's happened afterwards. So that's why I put the fish mass on the x-axis and frequency on the y-axis, because I think that is the response factor. Yes, Doug.
Were there particular nests that like did it seem like there were certain nests that really liked to just do a lot of short trips? Like somebody's like, all right, I want to Costco. Or did it seem kind of spread out, like, well, whatever they find.
We can look at this graph right now. So, well, I think there is actually, well, there might be some kind of correlation, but this is also like the average fish mass. So, it doesn't really mean like what kind of, this is on average. So if you're averaging things out, it doesn't make like mean that many things, but it would be interesting for me to go back to my raw data to look at like, oh, like if one nest is bringing back more bigger fish than the other nest. So I will maybe look at that later in fall. Yeah. Yes. Oh, they actually like they sink. We see them like sinking on water. So like a couple of guillemots would travel this like at the same time back, but we think that's like maybe to confuse like predators or like gulls because they would try to steal their food. And yeah, and we also found there's one nest, like the Halifor, the outlier one. I found that one. So there's like two nests that's close to each other, and the chicks from one nest actually moved into the other nest. So that could like mess up the data a little bit, but it's still like something that's interesting to notice. No, this different pairs of parents, and they feed them like through different holes, but the chicks would be like nesting in the same hole. Yes.
I think it's interesting to see, especially from the food, that a lot of these fishes are eating are red.
Yeah.
I'm wondering if you think that's random or if maybe there's something for red fish and if that's the case.
I think I don't know, but like you say, like because you're studying terns, you can see terns get all like the silverfish. Do you think that's like a visual cue for them? Because you're the bird expert here. Yeah, I think maybe they do have a preference for like not silverfish. Maybe. Yeah. Okay. Yeah, I was trying to mention like I'm thinking about that, but it's just like too anecdotal. Okay. Thank you.
All right, hello everybody. Thanks for sticking around for the end. I'm just gonna jump right in. So for my project, I was looking at assessing the impacts of investigator disturbance on the breeding success of black guillemots, so the same species as Abby's project. So just to start, what's a black guillemot? Abby already touched on a bunch of these aspects, but I want to re-highlight that these are circumpolar seabird species, meaning that they occupy the Arctic regions around the world. And so it's really interesting because the Isles of Shoals is actually the southernmost part of their breeding range, yet this population has only been studied and heavily monitored for 2 years, and little research has been done on this population in the southern region. So it's important to continue monitoring this local population as we are documenting the effects of climate change. I want to start with why ecologists even sample populations in the first place. And the obvious answer is to collect data and information on that population. And we call this demographic and ecological parameters. So things like growth rate or mortality rate or what these populations are feeding on. And these parameters can act as indicators of a population's health and status. And so ecologists often track these parameters over time to assess the changes in the population's health. And then that can be used to inform conservation and management decisions. So how do we even go about sampling a population? Well, in our case, when we were sampling guillemots, the first thing we had to do was actually find individuals. And that can actually be really tricky, especially because guillemots hide their nests in super well hidden rock crevices that are really hard to access. We also had to handle and observe individuals. So handling was required to collect growth rate data, and observing, you can collect data on behavioral patterns, so things like feeding frequency. And we also added bands and extracted feathers from these individuals. And so all of these activities that come with sampling guillemots can have potential impacts on that population. And so, for example, we might be altering the behavioral patterns of individuals. So guillemots might see human presence as a threat or potential predator. And instead of incubating their eggs or doing normal foraging behavior, they might prioritize more of that time to be vigilant on the human or on our presence. And so this is kind of acting as like a short-term survival response to our presence. And similar studies have been found on these changes in behavioral patterns in species like the common tern and the short-tailed shearwater when in the presence of humans. We could also be increasing the stress or energy expenditure of these individuals. So as we're handling these guillemot chicks, they see us as a potential risk and are going to try to squirm away and escape. And so another similar study that Elliott et al. Looked at was how there was increased levels of corticosterone, which is a stress hormone, in common murres from adding geolocators to their legs. And so all of these impacts are basically a product of what we call investigator disturbance, which is defined in a paper as all activities affecting individual birds or nests. So for my particular project in this study, there are a few activities that we can consider to be investigator disturbance. So the visiting of nest sites, the handling and banding of young, as well as the marking of nests. And so investigator disturbance that's caused from sampling a population can actually have a confounding effect on the exact biological parameters like growth rate or mortality that the researcher is interested in. And so the question you guys might all be asking is it even worth doing the research if you have the potential to, one, add bias to the data and confound your results, or threaten the exact population that you're trying to learn about? And so that kind of leads me to my question of how will different levels of investigator disturbance affect the body condition and feeding frequency of black guillemot chicks on the Isles of Shoals. And so we can expect to see that an increase in investigator disturbance will negatively affect body condition and feeding frequency of black guillemot chicks. And to kind of visualize that, I've set up a little diagram, a little plot here where we have the handling time, which is the time spent handling a chick. And that's used to collect growth measurements on the x-axis, and on the y-axis, we have body condition, which is a measure of how healthy that chick is. And so we would expect that as we handle these chicks for longer periods of time, their body condition would be lower. And so let's take a look at our study site. So you see each hot pink point is an individual guillemot nest, and you can observe that they're all kind of clustered in groups. And we call the grouping of these nests a neighborhood. And so there's a few neighborhoods both on Appledore Island and the neighboring island Smutty Nose. And on Appledore, we were looking at 20 occupied nest sites. 15 of those nest sites hatched at least one chick, and we were able to measure 17 individuals. Whereas Smutty Nose, we were looking at 15 occupied nest sites, and 13 of those nest sites hatched at least one chick, and we were able to measure 12 individuals. And so here's an example of what a guillemot neighborhood might look like. And the interesting thing that we observed was that was basically how shy these birds were. So when humans arrive at a neighborhood, we were consistently observing that all of the guillemots in that neighborhood were fleeing their nests and heading towards the water in hopes that us, the potential predator, wouldn't find their nest. And so this observation kind of led me to understand that human presence causes a neighborhood level disturbance rather than a specific nest level disturbance. And that kind of informed my experimental design, where I set up two different treatment groups: a high disturbance group, which is the neighborhoods in the red square, and a low disturbance group, which is the neighborhoods in that blue box. And so for high disturbance, we were visiting those neighborhoods 2 times per week and sampling chicks every visit during the nestling period. Whereas for low disturbance sites, we were visiting only 1 time per week, and we were only sampling chicks once to try to get a terminal measurement near the end of that nestling period. And so we collected a lot of different measurements when out in the field. A few of these include body measurements, so the bill length, wing length, and body mass, but also keeping track of time. And this is really important. So the time spent at the neighborhood, the nest, and the time spent handling chicks, because this is going to be our measure of investigator disturbance. We also banded every chick that we could get our hands on. And this helps us distinguish between individuals. And this is especially helpful if you have a nest with 2 guillemot chicks, so you know which one you're measuring. We also collected poop samples and feather samples. And lastly, we set up cameras to try to get behavioral data on feeding frequencies of these birds. And so field work was definitely a highlight of my summer. I loved being out on the rocks looking for guillemot nests, but it's definitely a challenge. It's no easy task. You really have to stay organized, and it's not a guarantee that every time you visit a neighborhood, you're going to get every chick just because of how well hidden and sneaky these guillemot chicks can be. And so once I had all of my data collected, I needed to find a way to measure the body condition of each individual. And one way I did this was by determining whether the weights of those chicks were above the average weight or below the average weight of the population. But to get that average weight, I first needed to generate a growth curve of the guillemots that I sampled. And so that looks like this, where we have the age of chicks in days on the x-axis and the weight of chicks in grams on the y-axis. And so you're going to see a bunch of scatterings of points, and each point is a chick that was measured throughout the season. And so that looks like this. There's definitely a strong correlation, which we would expect, but it's interesting to note that after around 21 days of age, you can see those weights start to level off, and we call that a growth asymptote. And the main indicator is that this growth curve is not a linear growth for guillemots. Instead, I used a sigmoid curve, which is also known as an S-shaped curve, to model the data, and that looks something like this. And you can see that this model is a really strong fit if you look at that R² value of the data that I sampled. So for this plot, you're going to see a few less points here, and that's because I'm keeping that same model fit, but I'm only taking the final individual measurements of each chick and graphing those. So instead of before where we had every measurement collected over the season, these are all separate individuals. And what you can do is you can take all of those individuals with weights above that fitted curve, and those are going to have weights that are above our predicted value or above the average of the population that I sampled. And then all of the chicks with weights below that fitted curve have weights that are below the average. And you can measure this by basically taking the distance from that observed weight point to that model. And that is what we call a residual. And that's what I'm going to be using as a measure for body condition, where positive residuals are healthier chicks and negative residuals are less healthy. And so here's the moment you all have been waiting for. Does investigator disturbance actually have an impact on the body condition of the black guillemot chicks on the Owls of Shoals? And so to orient yourself, we have cumulative handling time, which is the total time spent handling the chick throughout the season, not including that time spent during the final sample, on the x-axis, and then the weight residual on the y-axis, where positive residuals indicate above average weights and negative indicate below average. And you can see that our cumulative handling time ranges from around 0 minutes to over 100 minutes. And it makes sense that all of the points in blue have a handling time of 0. And this is because these are the individuals in our low disturbance group. And so these were never handled before the final measurement was taken. Whereas the individuals colored in red are kind of a range between 25 minutes of handling to over 100, depending on how often we could handle them. And collect data. And so that if you look at this R value, which is the correlation coefficient, you can see there's a really weak negative correlation between the residuals of the birds and the cumulative handling time. But if you look at the P value, you can see that that correlation is not a significant relationship. So the big takeaway from this graph is that there is not a statistically significant relationship between the time spent handling a chick and the body condition, which I thought was interesting. Now, I just want to point out, I tried really hard to see if there was any relationship between different measures of investigator disturbance. So not only can you look at cumulative handling time, but I also analyzed different explanatory variables, things like cumulative time spent at the nest, as well as the neighborhood visit frequency. And I just wanted to point out that I just wanted to see if these explanatory variables maybe had a stronger effect on the residuals rather than just the handling time. But after looking at all of these variables, I continued to see no significant relationships with any of them. And so I also looked at investigator disturbance and how that affects the feeding frequency, but I don't really have a ton of time to get into the main results of that. The big finding, though, and the main key takeaway of that kind of data was that investigator disturbance also had no significant effect on the feeding frequency of black guillemots. And so basically, these findings were really well supported in a lot of different studies that looked at different seabird species. And so you don't have to read all of this, but this is just evidence that there's finding similar trends with penguins, petrels, as well as the very similar alcid species, the Atlantic puffin. And so the main conclusion here is that the handling of black guillemot chicks for sampling does not have a significant effect on their body condition or feeding frequency. And this is great if you're a researcher, because it means, I want to go back to this slide again, because this conclusion really means that we can continue to sample and study this unique population on the Isles of Shoals without, and hopefully contributing to a long-term data set and monitoring project without worrying that we're confounding our results or threatening the population. However, it doesn't just end here. I'm still interested in exploring different angles of investigator disturbance on black guillemot populations. And one way I'm particularly interested in doing this is by, so just kind of set you up, we were only looking at how investigator disturbance impacted the nestling period when the chicks were alive. But I'm really curious if there's a stronger effect on investigator disturbance on the body condition of these birds or on the breeding success of these birds if we looked at how investigator disturbance impacts the incubation period or earlier on in the breeding season. So if we're disturbing them when they're incubating rather than when the chicks are born, will we see higher rates of nest abandonment or lower rates of nest reoccupancy in those following years? And I'm also interested in exploring other metrics of black guillemot fitness to compare with investigator disturbance. So things like stress hormones, like that corticosterone, and maybe the percent of successfully fledged chicks as a measure of how successful the breeding population was that year. So to wrap up, I just want to thank so many wonderful, supportive people. The whole team was needed to put this together. Specifically, Dr. Liz Craig and Gemma Klukas and my seabird partner, Abby. The whole Surge team, shout out to you guys for helping me out. And Mike Siegler and Rebecca Atkins, who are there every second of my time, just helping me with statistical analysis. And whether that's just like help from field work and collecting chicks, I want to shout out to all those helpful people out in the field with us, or just putting food on our plates every day from all of the staff. So thank you guys. Yeah, Doug. Yeah, that's a good question. My dad is actually an ethics professor and he teaches environmental ethics. So I'm always really interested in like the ethics of scientific research. But when I got here, I literally had no clue what I wanted to do. I didn't even know what species of seabird I wanted to study. And Willow kind of came out earlier on in the season and showed us these guillemot neighborhoods. And we kind of were just doing some observations to see what interested us. And I was really interested with how shy these birds were and how every time we approached them, they would immediately fly away compared to the gulls that are mostly everywhere on this island who would just try to attack you when you get near their nests. And so I wanted to know like how, like what the effects are of disturbing them, especially because they're such a shy species. Yeah. Not black guillemots, but they did us in the intro. I kind of talked about how they looked at higher levels of corticosterone in the common mirror. Oh, I'm not sure they tested for that in those studies. I think they were just looking at other factors. Yeah. Yeah, Morgan. Definitely. I think a lot of these studies that I was looking at and looking into investigator disturbance effects, a lot of them found more significant results on negative effects of the seabirds during that incubation period. And so I'm really interested in like if we disturb these birds to collect incubation data earlier on, how that might affect like the abandonment rates. And so I'm really interested in seeing if that is a stronger effect on the breeding population. rather than just handling. Yeah. Yes.
I don't know if that has any impact on it, but maybe on chicks survival.
Yeah, that's definitely something interesting to look at. One thing I want to add to that is like when we enter a neighborhood, we're obviously moving around a lot and like obviously hovering over these nests for long periods of time. And so I'm also curious if like our presence is kind of affecting these animals when we're not there, because we could be attracting like predators like gulls or like rodents to like come to these hot spots that we're kind of constantly visiting. And so there could be higher rates of like kleptoparasitism or stealing of prey just because they know where these nests are now. I'm not sure about that, but that would be something interesting to look at. Yes. Sorry, can you repeat that question? Yeah, that's a great question. So the question is about how I knew that there were chicks and not eggs during those different times of disturbance versus incubation and nestling. And so obviously not all of these eggs hatch at the same exact time. And so there's going to be some overlap between like causing disturbance during that preliminary incubation period until they hatch when some of the other nests in that neighborhood had already hatched. And so I'm also interested to see, because we have some data during disturbance during incubation periods too, to see if those sites had higher nest abandonments. And I actually, just based on like preliminary looking at the data, it seems like the ones that had later hatch dates had higher rates of nest abandonment, but I'll have to look at the data more carefully. Yeah. I think that's a myth. I think I think just because of handling, like birds are tend to be longer lived, especially seabirds tend to be longer lived in the bird world, and so they often put a lot of work into like caring for their young. And so they're not gonna like just abandon their nests just because we handled them. They're gonna put a lot of effort into trying to keep their young alive. And so I don't think that handling them in any way is gonna affect just abandonments rather than the other factors of success. Yeah. Wolfgar?
So I remember reading a paper about how like experience other hemisphere, birds and sometimes firefly prioritize their own safety groups to get when it comes to predators and different. How have you seen the video around? Do you know whether or not they're more focused on chick success or willing to risk themselves to a predator like yourself more? Or are they thinking like, all right, I would rather live and like make another one the next year and so flee in the way you have? Or is that like clean behavior and then trying to attract you away?
I definitely think it is a form of distraction, and like because they are so careful about placing their nests in such well hidden spots, I think that's probably the best bet for them to just distract you away from their nest in hopes that the predator doesn't find it. But also because they are longer lived birds, I think that they would prioritize their own, like especially earlier on in the breeding season, they're going to prioritize their own survival rather than their chick survival, because there's possibilities of re-nesting, especially if it's earlier on in that incubation period. Yeah.
We have lunch now until 1:15. We'll convene here at 1:30 for the intertidal ecology monitoring discussion. And let's just get one more round of applause for
So for our visitors that have not eaten a meal, you don't have to go before, you'll see that it's kind of a buffet family style dining experience. So several of them are probably helpful to help get the kitchen the food out. We'll set it up on these tables and then folks will just line up here, kind of plate, remote, and go on. And all the food will be marked that they have right here. So take a look at what they're eating. Goodbye.
The students, if we could just. Oh. You start sharing videos. Now they can see us in here. You can see that right here. I'll get rid of this because it's not important. And here is what you need. And when you're trying to advance your slides, you can just click or press the space bar. It should work. It's not us, that's not them. I feel like you can see the humidity of this room. Is that all right? What did you say? Oh, I said I feel like you can see the humidity of this room. Oh, yeah. Right. Okay.
Get in our seats, we can get back to business. Grab a water, grab a coffee, it's the old time. Coming in? You doing this? Right. Yeah. Thank you. We got that one shut. All right, so we're transitioning into the afternoon. Hope everybody's got a nice full belly. Wait, do I need to do anything with this? All right, good. Sorry. So the afternoon session starts off with our intertidal ecology team. For a little bit of context, this is Shoals Marine Lab's longest running monitoring project. It was a class project early on in the early days of the lab, and then it's transitioned into a undergraduate research internship project. It's 40 plus years worth of data from the intertidal all around Appledore Island. So you're going to hear from our team of undergraduate researchers from this summer. They have been led by a team of mentors this summer, including Drs. Kyla Bennis and Lauren Pandori, who are here with us today. And then in addition, Taylor Lindsay is a PhD student from University of Rhode Island and a longtime Shoaler. She's not here with us today, nor is Jennifer Dykstra, who is a research associate professor at University of New Hampshire. So without further ado, Kyla and Lauren.
All right, distinguished guests, family and friends, both in person and online. Thank you so much for being here and supporting the Rocky Intertidal interns in presenting their research project. Unlike some of the surges, which are on the island for 12 weeks, 10 weeks, 10 weeks this year, the intertidal internship takes place over an abridged 3-week span. And it is a frantic 3 weeks. So they start by arriving in Appledore Island and immediately going into the field and going to search for all the different algae and invertebrates that are on the island, learning to identify everything down to the species level, and then proceeding to spend every morning and every evening in the field for about 2 weeks, conducting surveys, 3 different types of surveys that have been conducted over time on this island. And then just when you think that you get to take a break after doing so much field work, then we shift into the analysis and data management and presentation part of the internship, where in a period of about 72 hours, the interns learn the statistical programming language R, and then do all of their analyses and data wrangling and figure making in that program. And then they spend another frantic 48 hours making their presentation. So I think, like, when I think about this internship, I think about the really intensive time and attention to detail and amount of heart and soul that the interns pour into their work. And there's always like a really wonderful kind of culmination of that at the end, which is their presentation. So keep all of that in mind as you like hear them talk today, that they've been through like so much and become such experts in a really short amount of time. And now Kyla will tell us a little bit about the history of the program and the internship.
Yeah. As Dave alluded to, this is a very long data set. In the 1970s, the first director had the foresight to establish 29 permanent locations around the island where students could return year after year to record the marine life occurring in the Rocky Intertidal. And for several decades after that, the students in the classes were assigned a transect that they would study over their entire time here at Shoals, and the faculty would collect their data sheets and store them in filing cabinets. In 2011, the Marine Lab decided to turn it into an internship, recognizing the importance and the value of such a long data set so that young scientists could be trained and the data could be ensure quality and accuracy of the data. In 2018, the lab hosted a data science surge, and that person gathered all of the historic data, combined it digitally, cleaned it up, and put it out on the internet. It's now publicly available for anyone who would like to explore that data set. So 2023 is the 41st year that we have recorded, and that will eventually be published online, led by myself, Lauren, Jen, and Taylor. And so the data seen today, we really want to emphasize, represents the collective effort of the faculty and staff that have preceded us, as well as hundreds of students over the last 4 decades. We're pretty sure this is the longest continuous record of intertidal biodiversity in the Gulf of Maine, and it couldn't be possible without that effort. So without further ado, this year's intertidal surges.
Hi, I'm Danielle.
I'm Tess.
Hi, I'm Ellie.
And I'm Yusuf.
And today we'll be talking about the relationships between algal canopy and invertebrates. We came up with this topic as we slid on seaweed while we explored the intertidal. So what is the intertidal? It is a zone between the lowest low tide and the highest high tide. The invertebrates in this area face environmental challenges due to the coming and going of the tide. So how many species do you think are in the intertidal zone? Feel free to shout it out.
Shout it out.
Well, from just looking at the slide, you might say there isn't much, or you might say there's a lot for some reason. However, when you look under the canopy, there's a lot more than some might expect. As part of the survey, about 58 species have been recorded over the years. So here are 3 canopy species that we use for our analysis. On the left is Ascophyllum notisum. In the middle is Fucus spiralis. And on the right is Fucus vesiculosus. All these are brown algae and are pretty common in the intertidal. Sessile invertebrates are directly attached to the substrate. Some examples of them are Mytilus edulis or blue mussel, semibalanus balanoides or barnacles. These can be found under the canopy or on their own, since these species are adapted to avoid desiccation. Botulus slosheri, on the other hand, are at higher risk of desiccation and thus found in the canopy. In regards to mobile invertebrates, we found a lot of crabs in the canopy, especially Carcinus manus. Snails like Litorina obtusata are also very common. Sea urchins like Strongylos centrotus, Jovachiensis are more rare. And so why should we care about the intertidal? Well, it supports aquaculture, the rocks protect against shore erosion, and there is an immense variety of species that call the intertidal home and depend on it. Not to mention, it can be a whole lot of fun. So the canopy is essential for the survival of many invertebrates that rely on it. It protects them from the harsh conditions during low tide, like the heat, UV radiation, desiccation, and predation.
So what is the goal of this project? Not only is the goal of this project to contribute to a 40-year-old survey of and database of intertidal species, to explore the relationship between algal canopy cover and invertebrate abundance, species richness, and community composition. So these are a few of the hypotheses that we came up with. The first one is canopy cover will display a unimodal relationship with both mobile and sessile invertebrates in abundance and species richness. So the figure below demonstrates a unimodal shape. And on the far left, we believe that there will be less invertebrates because there is no cover. If there's no cover, then they are not protected by abiotic factors such as waves and radiation and predators. That's a biotic factor. And to the far right, the other extreme is that if there is too much canopy cover, then they might be outcompeted for space and food. Oh, and our other hypothesis is that as algal canopy cover changes, so does the invertebrate community composition. This means that with different levels of canopy cover, we're expecting different species to be in those areas.
Okay. So for the purposes of our study, we chose to use the Shoals protocol data to explore our research question. And in the Shoals protocol, there are originally 29 transects. However, for this study, we only surveyed these 6 shown because they were the most widely studied over the years. And of these transect sites, we have transects 5, 7, and 26, which represent the sheltered side, which is on the west side of the island, protected from the waves. And then we also have transects 15, 20, and 22 on the exposed, which face the ocean, so it results in a lot of wave action. So at each transect, we assess the percentages of each canopy species, then move the canopy aside to count and take percentages of the species on the subcanopy. So for example, pictured on the left is our canopy assessment, which is filled with Ascophyllum, in which we would say 100% coverage. And then afterwards, we would move it to the side and typically find a surplus of species like green and red algae, mobile and sessile invertebrates, pictured on the right. So overall, we surveyed 3 quadrats at every level, ranging from 1 to 13 of the specific transect. And after data collection, we basically all became coding professionals, thanks to our mentors. With our new scales, we ran regressions that compared mobile and sessile invertebrate abundance and richness to canopy cover across exposure. And additionally, we ran a PCA to examine invertebrate composition relative to exposure and canopy cover. So here are the results. Pictured is our first figure where we have algal canopy cover measured in percent cover on the x-axis. And then on the y-axis, we have mobile invertebrate abundance. On the left are the exposed sides with the pink, and on the right is the sheltered sides in the blue. And just keeping in mind that every dot represents the average of mobile invertebrates on every transect per year. So although we found no strong relationship of invertebrate abundance and canopy cover on either exposed or sheltered in this graph, we still made some pretty great observations. The first being how the algal percent cover is much higher in the sheltered transects, which is expected as canopy species like Ascophyllum can grow better without the harsh wave action in the exposed. Additionally, we found a greater range and variation of invertebrate abundance on the exposed side. And lastly, we see that there's no clear trend in the sheltered site. However, on the exposed, there is an observed unimodal trend that Yusuf talked about in the beginning, in which we see a peak around 35% that produced the highest amount of mobile invertebrates. So similar to the previous graph, we are comparing algal canopy cover with invertebrate abundance, but this time we're looking at sessile invertebrates, which are animals without backbones that are attached to the rocky intertidal. On the sheltered side, we don't see a clear trend again, but on the exposed side, there is a statistically significant relationship that supports the unimodal distribution, in which the peak is around 20%. This peak again represents the quote unquote sweet spot for the perfect amount of shelter, moisture, and space. And lastly, we observed how more sessile invertebrates are found on the exposed side, which makes sense as some sessile species like mussels and barnacles have strong attachments to rocks and would in turn be abundant in high wave action.
Okay, so this graph represents the species richness of mobile inverts in comparison to algal canopy cover. As you can see, neither of the exposed or shelter side graphs have significant results in relation to canopy cover, but we still see some useful trends. For example, both graphs show a unimodal-like trend in which the exposed side peaks around 35%, while the shelter side peaks around 50%. We also see that the sheltered transects support a greater diversity of mobile inverts, and this is most likely caused by less wave action, like Ellie mentioned, resulting in a safer environment for those mobile inverts. And this graph represents the species richness of sessile inverts in comparison to algal canopy cover. And in the exposed transects, we see a unimodal trend with a peak around 35%, which is very similar to what we saw with the mobile invertebrates in the last slide. Furthermore, on the shelter side, we see higher canopy abundance, but lower sessile invert richness. And another very interesting trend that we see is that it's not a unimodal shape, it's kind of like an inverted unimodal shape, which is the exact opposite of what we thought we would see. And another thing to note is that this is a significant trend.
So now shifting gears to the community composition, we ran a principal component analysis to assess which species are driving differences in sessile invertebrate community composition. So a principal component analysis condenses multidimensional data into these principal components that represent the directions where there's the most variance. So the closer 2 points are together on the plot, the more similar those transects are with respect to whatever data was put into the analysis. In this case, that data was sessile invertebrate abundance. Also on the plot, the transects are labeled based on their exposure. So the blue ones are sheltered and then the red ones are exposed. And from looking at this plot, we can see that exposed transects seem to support different sessile invertebrate communities than the sheltered ones. Anna Permanova revealed that exposure did have a statistically significant effect on sessile invertebrate community composition. What we can also see is that exposed sessile invertebrate communities are driven by barnacles and mussels, whereas the sheltered ones are driven by a more diverse assemblage of sessile invertebrates. And this is what we might expect, since we know that barnacles and mussels are adapted to thrive in more wave-exposed environments, whereas other sessile invertebrates like Botrytis slosheri are much more sensitive to wave exposure. So this is a similar principal component analysis, but this time the data that was input was mobile invertebrate abundance. And Permanova revealed that exposure also had a significant impact on mobile invertebrate community composition. But if we look at the plot, we can see that the clustering within transects is experiencing similar levels of exposure is less clear than it was for the sessile invertebrates. So this suggests that differences in wave exposure might have a larger impact on sessile invertebrate communities than on mobile ones. Coming back to the sessile invertebrates, this is the same PCA as the first one that we saw, just this time the transects are labeled based on the amount of algal canopy cover that they experience. Looking at the plot, we can see that there is some grouping within transects that are experiencing similar levels of algal canopy cover. And that was confirmed by Permanova. Algal canopy cover did have a statistically significant impact on sessile invertebrate community composition. And then finally, for the mobile invertebrates, we can see that there's more clear clustering within transects experiencing similar levels of algal canopy cover than there was for the sessile invertebrates. This suggests that algal canopy cover may be more important for driving mobile invertebrate community composition than it is for the sessile invertebrates. So just to summarize some of our key results, we found that on sheltered shores, there was greater canopy cover. There were less abundant sessile invertebrate assemblages, but greater mobile invertebrate richness when compared to the exposed shores. Nonetheless, we did find some unimodal patterns between canopy cover and invertebrate abundance and species richness, but most of these weren't significant, so we can't say that our hypothesis was supported, but it'd still be a really interesting relationship to explore further. And in terms of community composition, we found that sessile invertebrate communities differed most across exposure levels, while the mobile invertebrate communities differed most across different levels of algal canopy cover.
So our last figure shows that canopy cover is absolutely increasing over time. And there has been recent studies that have shown that ascophyllum length and other key canopy species are increasing due to ocean acidification and global warming. So our results suggest that perhaps composition, abundance, and richness could change with this increase in canopy cover. However, there are a lot of components and interactions to consider within that. So we can't solely place canopy cover on invertebrate abundance or richness as we hypothesized. So with this, we encourage more long-term surveys exploring different components in addition to canopy cover.
So some things we'd like to see further investigated in the future are, like Ellie said, the length and cover of ascophyllum in regards to ocean acidification, more on the impact of canopy cover on invertebrates, and we'd like to see more long-term surveys established in the Gulf of Maine.
We'd love to give a huge thanks to our mentors, Dr. Kyla Bennis, Dr. Lauren Pandori, Dr. Jen Dykstra, and Taylor Lindsay for guiding us through and allowing us to have be a part of such an important project. Unfortunately, Taylor and Jen were not able to join us today, but if you're watching, thank you guys so much. And we'd also like to thank Dr. David Buck for bringing us to the island, the other surges for supporting us through everything, shout out K-Norts, and to the entire Shoals family for making this experience so amazing. And to the Best Intertidal team, and thank you to our pet crab who helped us through it all.
Yeah, it's a really nice thought. I think update, I agree that more monitoring is great as well. As well as
One thing we were really interested in also was looking at like how ascophyllum length changes over time. So I think it'd be really interesting to look at, how does warming affect ascophyllum length, or even any of the other canopy species like in a laboratory setting, because that could also affect the invertebrates and other downstream.
Looking at different individual transects as well, It is really cool. We pulled up some graphs for that. We're still looking at that data, but that's also another topic that
Do you think after doing this program that you guys will find interesting with your pilot studies in the future? When you guys personally been very inspired by this?
I really like snails and I hope to study them in the future. So I might end up in the intertidal in the future one day, and I already studied the intertidal at home, so.
For me personally, this is like my third year working on intertidal surveys. In the past, I've worked specifically with bivalve surveys and like formulating the protocols and stuff like that. So I think this really helped with my interest more though, because I didn't realize how many different species were under the algae canopy, and it just like sparked a new interest.
Yeah, for me, I've always loved the New England coast. I'm from Canada, but we vacationed here a lot, and it was really special to come here and learn about all the species. And I definitely love to look out here if I can one day, and or intertidal elsewhere. Yeah, maybe full circle. But yeah, same here.
Thank you.
Really great presentation, very cool data, really work.
Thank you.
One of the things I thought was really interesting from my perspective was the graphic that looked at sheltered versus exposed and the species that were driving the differences in the PCA. All of the sheltered were invasive, most of them are invasive species. Can you talk a little bit about your observations? Like when you went to the exposed side, did you not see them? Or can you talk a little bit about that? I'm very curious about that graphic.
Yeah, that one. On the exposed side, there was like a lot of barnacles, and they're also like really big mussel beds. So I think in general, we were seeing less like inverts, I would say, especially like the bryozoans and stuff were a lot more on the sheltered side. So I didn't like visually notice any differences in terms of like invasive species, but there was really the shift where on the exposed side, we have a lot of barnacles and a lot of mussel beds too. Yeah.
So another question I had was the protocol. So if you find organisms living on top of other organisms, like Midas sedulus is, you know, there's lots of foul organisms on that. How would you deal with that in the data?
They would both be counted as like percent coverage. For barnacles, there's a surplus of them, so we have to like count them individually. Like some barnacles would be on top of the mussels.
I guess we also kind of like in the Shoals survey, we first survey the canopy like without moving anything to see like the percent cover, and then we move things to look at what's attached to the rock, and then we estimate percent cover of the things that are attached. So like barnacles and mussels would go into what we call the primary layer. Yeah, and so we measure their percent cover and then also count and size them.
Yeah. So you don't count things that are on top of nothing.
We do. Everything in there gets counted.
Okay, we count them. We count them if it's not like coverage.
Like we count them all at once. Yeah. Yeah.
Great. Thank you.
All right, we have the privilege of wrapping up the research symposium today by introducing the shortest and most intense version of the different flavors of research opportunities that are provided to students at Shoals. So the students in the research apprenticeship program take at least one class on the island sometime in the summer or in a previous year that affords them the opportunity to get their feet wet, usually in the period of 3 days to a week, in the context of another course. And that could be field animal behavior, or it could be marine parasitology and disease. These students then get the opportunity to spend an additional 2 weeks on island taking a class called Research in Biology, where they have those 2 weeks to spend pursuing from start to finish a project of their own conception and design. And they take it from figuring out and refining a research question, usually within the first 48 hours of that time, all the way through analysis, visualization, and interpretation, and communication. So we've been delighted to work with this year's group. They've been getting up early in the morning, going to bed late at night, waking up in the middle of the night, combing through hours of video or thousands of rows of observations that they made in the field, learning and using an entirely new computing environment to some of them, the R environment, which they'll all show you their visualizations in. And we couldn't be prouder of the way that they have tackled this challenge. So they have worked not only impressively on ambitious topics, but the way that they have worked has been equally, if not more impressive. They've shown tenacity, camaraderie, curiosity, good humor, and these are all sort of the key ingredients in, I think, of a lot of our minds that it takes to really, decide to pursue a career, or at least some next steps beyond a 2-week experience in this sort of fun, magical world of marine science. So the format of their talks is they will give a shorter version of an oral conference presentation. They'll have about 8 minutes to speak with 2 minutes for questions. And first up, oh, and I forgot to say, Rebecca and I got to co-advise these students together. And I just want to give, in addition to shouting out the amazingness of the students, a shout out to Rebecca, who's been advising these students, the Surge students, and also working full-time for NOAA in the background. So thank you.
So without further ado, we have Menange de Leon talking about the abundance and interactions of the anemones Diadumene lineata and Metridium senile on Appledore Islands.
Oh, okay. Hi, everybody. I'm Yaj, also known as Mimi around here. And I decided to focus on anemones. But why anemones? There's so many other species on this island, but why them? Well, if you see this beautiful place, it all started with my home country, St. Lucia. It's in the Caribbean. It's right next to Barbados, if you know where Barbados is. I would normally go snorkeling there. I would sometimes just be going for a run on the beach and somehow end up in the water because I'm really obsessed with marine life. And I would see a lot of different species like trumpetfish, coral, anemones, sea urchins, a lot of cool stuff. And I go into marine biology. I would do a lot of readings, listen to a lot of people speak about their what they're studying or their research. And I realized that every single time I was always obsessed with anemones or cnidarians and jellyfish. So I still have that obsession today. So I was so happy that I got to do research on anemones here. And my species of focus are Metridium senile and Diadumene lineata. I don't know if I'm pronouncing it right, but you can correct me later if you want. So Diadumene, well, Metridium is very, okay, I'm so sorry, I'm so nervous. Metridium is most commonly known as the frilled anemone, and it also has many other common names. These guys can be identified by their smooth column, which is usually brown, orange, or white. And they also grow 100 to 1,000 tentacles that are usually shorter compared to Diadumene, which is on the right, your left or your right. And Diadumene is also commonly known as the orange striped anemone. And they are usually olive green with yellow or orange stripes on their column. And sometimes I do see them with the white stripes. They usually grow 50 to 100 tentacles, and it's usually longer than Metridium. Okay, so I have a question for you guys. Which do you think is native and which do you think is non-native if you've been paying attention to the previous presentation? Anyone?
One on the right is invasive.
Did you know that already? Okay, and you are correct. So, yes. Native Metridium and non-native is Diadumne. Diadumne is native to Japan and the Western Pacific, and they usually, sorry, before I go there, Metridium is native to the Northern Hemisphere. They're both usually found in the rocky intertidal or shallower water that contains stuff like mussels and oysters, and they prefer habitats with hard substrates that they could attach themselves to, like mussels. And they typically feed on zooplankton and epibenthos. And compared to Metridium, I learned that Diadumnate can tolerate a larger range of conditions. And because of this, I decided to ask a question. Compared to Metridium, is the abundance of Diadumnate greater in tide pools around Appledore Island? And so I came up with the hypothesis that Diadumnate will be more abundant across the tide pools since it tolerates a wider range of conditions compared to Metridium. So I decided to go ahead and test that out. And I picked 3 sites on the island to do some sampling and collect some data. The first site is Laurel's Ledge, it's all the way to the north, and Transect 5, which is right below it, and Half Tide Ledges, which is all the way to the south. And when sampling, I sampled at low tide so I could be able to see the species much better. And I sampled in each three pools in each zone. So the first one was the rocky zone, second is ascophyllum, and the red zone is the red algae zone. And the ascophyllum zone usually has like brown algae, and yeah, the rocky zone is more exposed. And I did my data collection by using a diometer, where I would record the temperature and the dissolved oxygen in each of the pools. I also use a refractometer to measure the salinity. And I used a transect tape to measure the length of the pools, and use a quadrat. I would throw a quadrat into the pools depending on the size that I measured. And I would count the amount of species, each of the species underneath the quadrat, and I would use a ruler to measure the depth underneath those transects, those quadrats, sorry. And then going into my results. So first, I have the means of the 2 species, Diadumnate and Metridium, the abundance means, and they're very close, they're very similar. And I ran a statistical test to see if there's a difference, and there was no significant difference in the mean abundance across both sites. And then I decided to look at the 3 different zones, the abundance in all 3 different zones. I realized that in the ascophyllum zone, there was a high abundance of Metridium. In the rocky zone, I only discovered Diadumnate, and in the red zone, or the red algae zone, there was a very low amount, or yeah, a very small amount of each of the species. And then I decided to plot the abundance of both the species, trying to see if Diadumnate abundance affects the Metridium abundance, because it's not a non-native species. And I realized that there was, well, after running my statistical test, I realized that there was no significant relationship or effect of Diadumnate abundance on Metridium. But what I realized was interesting was that when there was 0 Diadumnate, the Metridium abundance was greater, and when there was 0 Metridium, there was more Diadumnate present. And I also tried to see if the average temperature, the salinity, and dissolved oxygen had any effects, and there was no effect of all these abiotic factors on their abundance. And lastly, I tried to see if the quadrat depth or the depth had any effect on their abundance, and realized that after running my test, which made me sad, no, no significant relationship or effect. But I did realize that a lot of the species that I found were more in like shallower areas or shallower waters. And so that brings me all the way back to my research question. Is the abundance of diatomnate greater? And the answer is no. And that could be due to my limited amount of data. I only had 2 weeks, actually a little bit less than that, because I picked my topic kind of like a little bit of a ways into the first week. And there were a lot of pools, like the red zone pools, like with the red algae closer to the water itself, the ocean, where there were no, a lot of them had no anemones at all, which made me sad because I really was so excited to see if there were any in there, and there were big pools. And for the future, I would like to do more of this and explore, do more data collection all around the island, collect more data from more pools. And I also I'm very interested in DNA and like how we can use DNA from anemones or species in the environment to find out what other species are present in their environment. So that's all that I have for you guys. And I just want to say thank you. Thank you to my wonderful professors, Rebecca and Carrie. Love you guys. Thanks for sticking with me through R. We've been doing this since last night. Went to bed at like 12 a.m. Right, yeah. And yeah, all my classmates and the intertitle folks, thank you so much for letting me go out there with you guys and see what's out there. And Shoals, love you guys. Thanks so much. All right. I think we have time for one question. Oh, raise your hand first. Are there other kinds of entities besides these two? I'm assuming there are. I'm not completely sure. I asked the intertidal folks what kind of anemones that are most commonly found. I didn't really ask them like what like all of the anemones or species are around here. But I'm assuming there are. I'm not sure. That's a question that we'll all need to discuss later. Yeah. But yeah. Thanks so much.
Thank you, Mimi. Our second presentation is from Alexander Luna. Alexander is going to talk to us about hagfish succeeding in river.
Hello. All right. So I chose to study hagfish for the past 2 weeks. Well, really for 4 weeks. I'm kind of becoming a hagfish guy, interestingly. But I know that not everyone is a hagfish guy. So I just want to start off with a question, and feel free to raise your hand for this. But how many of you guys know what a hagfish is? Okay, I feel like that's to be expected out here. So I'm gonna stump you guys. How many of you guys know what a hagfish does? All right, that's cheating. I'm lying. You've spent 2 weeks with these guys. Well, hagfish do a lot of this and this. And even though these are static images, if these were videos, it'd still be the same. They don't really do a lot. They spend a lot of their life sedentary, and this is true even in the real world. This is an example of a Pacific hagfish sitting outside of its burrow. And hagfish are a group of benthic, jawless fish, which means that they spend their most of their lives at the bottom of the ocean, kind of sitting around. There are opportunistic scavengers, which means that they wait for opportunities to find prey or wait for huge animals like whales to die and drop to the bottom where they can feast on their carcasses and feast inside of them. But as a result, they spend a lot of their life sitting, and they have many adaptations for this life of just sitting, which means that they have very low energy requirements as a baseline. And also, if you've ever heard of these guys, you might have known of their notorious ability to slime. And our Atlantic hagfish out here can be found at around 300 meters,
but they really do have a large range of depths, all the way from 10 meters to about 1,000. From my knowledge, a lot of the Pacific ones you can find closer to the surface, and those are the ones where you could find at like 10 meters, which is pretty interesting. Okay, so now we know about hagfish. How do we get circadian rhythm from there? And I just want to start out with a couple of baselines. So circadian rhythm, I feel like, as many of us know, is our biological clock that regulates and directs processes from the macro to the micro scale. And it usually is on a 24-hour cycle that has a lot to do with the light cycle as well, which is why it's 24 hours long. And how do we detect circadian rhythm? Well, circadian rhythm can be seen in patterns and changes in body temperature, metabolic rate, and activity cycles. And one example of this is in a naked mole rat, where you can see during a 12-hour dark period, their oxygen consumption rates spike up. And then during the daytime, it's quite low. And this sort of pattern that we see during night and day is evidence of circadian rhythm. But how do we get to hagfish from here? Well, studies have been done, believe it or not, in hagfish on circadian rhythm, although it's a group of 2 scientists. But they found that Pacific hagfish swim more at night, so they have nocturnal activity. And another study, although it wasn't focusing on circadian rhythm, found that this translates to their metabolic rate, where you can see in the steepness of the slope over the 2 dark periods, it's way steeper than it is during the daytime period. So hagfish are consuming more oxygen at night. But if you paid attention, you might notice that I've been talking a lot about Pacific hagfish. What about our Atlantic hagfish that we have around here? Well, there's no information on the circadian rhythm in Atlantic hagfish. So I set out as a pioneer and asked myself the question, how do Atlantic hagfish regulate their metabolic rate? And now, based on everything that I've been building up, the obvious first hypothesis is the circadian rhythm hypothesis, that these Atlantic hagfish have a circadian rhythm, and it follows a general trend that they're more active at night. So their metabolic rate will see an increase in the oxygen consumption during a nighttime period. And now another hypothesis that I have, an alternative one, is the stimulus hypothesis, which states that since hagfish have a very sedentary lifestyle with low energy requirements and long times between meals, it's energetically unfavorable for them to increase their metabolic rate unless it's absolutely necessary. So how do I go about testing this? I decided to use a box, a very special box, a respirometer. So that's that clear acrylic box on the right, which once the organism is placed inside, it is sealed off from the outside world. And this allows us to measure the oxygen concentrations within the box using an oxygen probe, and then also temperature to make sure that they're staying in a comfortable environment. And then there's also a way to inject oxygen into the system, since this is a static system and it's not very fancy. So you got to do it the old-fashioned way. But in order for my experiments, I needed to modify this to dark mode, which is where I covered the box in tape, because I wanted to limit the amount of variables that I was influencing on these hagfish. Because if they had a circadian rhythm, and at their environment at 300 meters where it's very dark, they likely would not be experiencing much light. So if there was a presence of a circadian rhythm, it would present itself when there's no stimulus provided to these hagfish in a completely dark environment. And thirdly, I used a computer setup and software to monitor the oxygen concentration in the tank, which becomes very important when I show you guys the graphs that they produce, where you can see a nighttime, daytime, and then night again. And you can see an overall trend in decrease of oxygen in the respirometer. But you may be wondering, what are these weird spikes and weird colorations on the graph? And don't worry, I will explain them. So in the green circle, you can see a spike of oxygen. Since we're working with a static system where we don't have any way to have oxygen manually or automatically enter the system, I have to go in and pump oxygen. So about every 4 hours, I have to pump oxygen into the system. And then I took the metabolic rate about 30 minutes afterwards in order to give them time to acclimate to this new oxygen that's been introduced into the system, and also so that way it wouldn't have any effect on the metabolic rate. Although research that has been done before has shown that hagfish don't really care if you inject water into the respirometer. They don't really show any sort of effect by that. So the dark areas are the regions that I sampled to figure out how fast they were consuming oxygen. Now, what about the results? So overall, the hagfish metabolic rate is extremely low. And I realized for many of us, what does this low value mean? And something that I looked up just this morning is that the human brain, and just the brain alone, consumes about 93 micromoles per gram per hour, which you can see all of this is less than 1. So our brains are working like 90 times harder than a hagfish. But this agrees with their burrowing adaptations in general ecology, because if they have such long times between meals and spend a lot of time sedentary, they don't really have a high energetic requirement. But as we can see for Hag2, this little guy has some very interesting things going on with his metabolic rate with a couple of high values. And this is likely due to its small size. And I just removed it out of the data pool for my graphs here. So you can see that there was no significant difference between the metabolic rates at day and night because of how similar they are. So what does this tell us about hagfish? Well, firstly, the low metabolic rate data corroborates with results from many, many papers when you take into account differences in species and then Atlantic versus Pacific hagfish. And this sort of study serves as a baseline for future studies on hagfish circadian rhythm and metabolic rate, and also studies on hagfish and their general ecological niche, where as they live in the sediment, you could look at bioturbation, climate change, and how these hagfish are affecting it with their energetic requirements and such. And then on the study itself, there's, as I've mentioned, there's no strong evidence for circadian cycles of activity in hagfish. But one interesting thing that I noted, and for future implications, is that when there were differences in the slope, which was very few and far between in the 6 hagfish that I measured, they normally happened near sunrise or near sunset, which has different implications for the general ecology, as much of the data for hagfish and energetic requirement estimates for populations have been done during the day. So if it turns out that these hagfish are more active at night and consuming more energy at night, this could mean that all of our math is wrong and all of our estimates are wrong. So the hagfish ecological community will be shocked if this turns out to be true, but more repetitions are necessary. And just asking future questions of, will Atlantic hagfish entrain into a day-night cycle like the Pacific ones did? And then also, will these patterns behavior continue with more trials? More than 6, maybe double, triple, yeah. So these are my references. We'll just go through that. And then acknowledgements, I'd like to thank everyone in RIB, all of my mentors during RIB, Carrie and Rebecca, and then also my mentors from IMBL, the previous class I took for this. Everyone at Shoals Marine Lab for helping me through this, and the staff for keeping us fed. You guys for listening to me spiel about hagfish for 8 minutes, and yeah. Yeah. The short answer is I don't believe so. The long answer is that we don't really have much knowledge on their reproduction. All of these hagfish, even though they do spend a lot of time at depth, they still have eyes, although they're covered. Some in Pacific, they're covered by skin, and in Atlantic species, they're covered by a thicker layer of muscle. So we know that they still can perceive light, but just necessarily, I don't think they experience much light where they are in their life in general, but more data will need to be collected on that, and yeah.
You might have heard the sentence where you said that he collected 6 hagfish worth of 24-hour trials with 4 hours oxygen changes in the river.
Are we? Did we put this in twice? No, good. There we go. Oh, I think we're out of order. Let me, hold on, temporary pause.
Technical malfunction, I'm sure it's.
No, it's
We need to go from her by copy pasted the wrong thing in. I have a feeling that we need your Gmail and we need the SML folders.
It's okay. Just gonna see if it's in here. Okay. No, we good.
There we go.
We're just gonna play it.
Yeah, okay, and then is the other one still pulled up? So
The
that
other one will immediately go into.
let's just copy paste them and it goes pretty fast. So
Okay. Copy.
Right above break. Right after that.
We're good. Still up and running. This is it the most updated version, or did you change your font version? Yeah. If anything's weird, just let me know.
Okay.
Okay, thank you for your patience. So right now we're going to listen to Morgan Lane discuss Loris argentatus and Loris marinus adult and juvenile distribution and behavior in littoral microhabitats on aquifer.
Again, I'm Morgan. Hi. You might be, what is Loris argentatus and what is Loris marinus? They're gulls. And there's a lot of them on this island. Many of you may know, even if you've been here for just a couple hours, there's significantly a lot of gulls on this island. And just to show what each of the species and age looks like, the two species that are on this island are herring and great blackback gulls. And herrings are the ones that have gray wings, and the great blackbacks are the ones that have black wings. And the juveniles, both are a little bit gray and whitish, but the juveniles of the great blackbacks are a lot more whiter than the herring juveniles. And they're both, or herring are more generalized foragers compared to the great blackbacks. And you may ask, what do they eat? It's not currently. Neither's working. Oh, just swipe it. Okay. Well, one, they eat crabs. Oh. Oh. Oh my God. I can do this. Crabs. Oh. Fish. Oh, I'm sorry, mussels. And herring specifically eat trash and compost, which isn't really a problem on this island because we contain them in sheds and little containers. But there is competition between two of the species. And great blackback gulls are a lot more aggressive because they're a lot larger as well, so they can tend to be more aggressive. And most of the aggressive interactions that happen on this island, the great blackback wins with the first medal. And they all feed in intertidal zones, but gulls tend to feed more inland because the great blackback gulls are more considered more seabird-esque because they feed more on the coastal areas, where herrings, you may see them at your local McDonald's. So a little bit about juveniles. They're
Look away. I have no idea why that decided to fall.
That's okay.
Yeah, no. Yeah, it's just a super fun moment for all of us. Okay, Okay, now you're good.
Hopefully. It's good now. Okay, sorry for the technical difficulties. But now a little bit about the juveniles. They're overall less efficient foragers because they don't have as much experience because they're younger. And they often go for less successful foraging areas because there's less competition there. And because of this, the mortality of the juveniles increases a lot, and it's pretty high. And they still to this day on Appledore, they're still calling to the parents and making little meeping noises for resources such as food and protection between the other gulls. And there is because of the chick season, there's a high competition between the parents and species. But how does this happen, or how does this affect the fledgling and juvenile season? So a little bit about the microhabitats that I will be looking at. This is a picture of Half-Tide Ledge, which is on the southern part of the island. And I split it up into 3 little sections, which rocky area, which is the dry rocks, the edge or intertidal zones, which is where the algae and wet rocks are, and the water, which is obviously where the ocean is. And I also looked at their behaviors. So I categorize them into 5 different behaviors. So I did resting, which is when they're kind of in a little, I call loaf mode. Preening, which is when they're cleaning themselves. Observing, which is when they're standing on their 2 feet. Foraging, which is when they're actively pecking their beak and heads into the water or seaweed. And calling, which is when the juveniles make the meeping noise or when the actual adults are squawking. And so this, all of this collectively helped me create questions and hypotheses. And my questions looked at what is the distribution between the species and age of within each microhabitat, and additionally, what behaviors are they portraying? And my hypotheses, I categorize them into microhabitats and behaviors, or I split them up into 2 different parts. And when herring and great blackback gulls are observed at low tide, there will be a higher abundance of great blackback adults and juveniles found in the favorable microhabitat, which is the edge. And herring adults and juveniles will be found on the rockier areas. And then behavior-wise, great blackback gulls adults will be found at a higher abundancy foraging, and then both the juveniles will be found making calling noises. And so I collected data at 2 different locations on Appledore, Laris Ledge and Appledore Ledge, and Appledore Ledge is also called Half-Tide Ledge, which I mentioned earlier. And I took count of the species and age, and I also took count of where they were within the microhabitats and what behavior they were exhibiting. And I analyzed all of this data in R Studio. Okay, so this is Laris Ledge. I'm going to run you down, I guess, my collecting of data. That's me standing on a rock. And this is when I look into the ocean, this is what I see. And okay. Okay, and I separated them into 3 different categories, which is 45 degrees to the left, 0 degrees, and 45 degrees to the right. And I took each of these snapshots of these degrees. I spent 15 minutes looking at each degree, and I took collection every 3 minutes and looking into what a 0 degree might look like. It's not working if I work to the space bar. I would look at I would count all of the gulls. Here it's kind of hard to see which species are, but when I use binoculars, it's a lot more easier. And because of the way I calculated all of this, for the microhabitats, I grouped them. So my n size or my sample size was 6, and my sample size for the behavior was 4. And a little bit now into my results section. I first will talk about the microhabitats. And each
I separated them each by their microhabitat. And I also labeled, as you can see, the AABC. So I looked at the how they overlapped each other. And this is what all of them look like. And I'm going to note really quickly here that in the edge habitat, there's significantly a lot more gulls that can be found. And overall, there was a lot more gulls that could be found over the edge compared to the rocky and waters microhabitat. And then now looking into the proportion of time of what they were actually doing. There was a lot more gulls observing, as you can see in the middle of the graph over there. And how does this support or not support my hypothesis? Looking a little bit back into the microhabitat hypothesis, there was significantly, in a little bit of an overview, there was significantly a higher abundance of species on the edge. And both the species preferred the edge habitat, but there was minimal competition. And this could be also possibly due because of the fledging season and it's not chick season anymore. And looking into the behavioral hypothesis, there's overall more species found spending more of their time observing rather than any other behavior. And the time spent foraging is very small comparatively to the observing, which is what I originally hypothesized. And foraging, they may be foraging at differently. Rather than at low tide, they may be foraging at high tide or mid tide, which I only focused on low tide. And some further research, because of the abundancy of how many herring gulls there were, herring gull juveniles, I would want to focus more on how their competition is between the actual juveniles. And a quick thank you to everybody. And yeah, Kelly.
After living on the goal on all summer, getting really meetings closer, look at the past 2 weeks. What is your favorite goal behavior and or favorite goal fact?
The question is for people at Zoom, what is my favorite goal behavior or fact that I've observed within the 2 weeks of doing this research? I really like the meeping noise that the juveniles make. I think it's funny. And just like the different calls that they make are weird. And like how the adults or the juveniles, when they're asking for the adults for food and they come up really close and the adults just always look annoyed. But yeah. But yeah. Thank you.
Actually. You're just using arrows or something.
Well, also, when you click on it, you can do the. Okay. We're just gonna go with it.
You're gonna recommend arrows or?
I'm gonna recommend spacebar. Although if she clicks on it, then try the arrows, but only. So do the spacebar, and if the spacebar is giving you issues, just do that.
And for audio playback?
You can just look and click.
Oh, sick.
Yes.
All right, our next presentation is Mia Suresh. She's going to talk to us about parameters and concepts of call recognition in great catbirds.
Thank you for that little intro. Okay, so I'm just going to start off with like a broad idea of what signal detection theory is. So you can imagine like the environment has a lot of different sounds and animals need to pick out what the relevant sounds are. And this is especially important to territorial birds like the catbird. So there are kind of 4 outcomes, and I'm going to sort of walk you through this graph right here. But there's correct detection, which is a hit. There's correct rejection, misdetection, which is a miss, and then false alarm. So if you look at like the top figure, there's 2 kind of sounds happening. And the one on the right is the sound that is like specific to that animal, the one that they want to detect.
And that line in the center is the threshold. So it's like what they're deciding to kind of scan for, how they decide like what they're going to take in and ignore. And then the shaded part on the right is a hit. So that's a positive outcome. And then the part on the left is a miss, which is a negative outcome, but it's a very small negative outcome. And then in the second figure below that, it's the same thing. The shaded part on the left is, well, that entire figure is going to be the sound that you don't want to detect. So the part on the left is correct rejection. And then the part on the right is going to be your false alarms. So basically the idea of this is to maximize positive outcomes while minimizing negative outcomes. So how does this apply to catbirds? Well, catbirds are territorial birds, and there are many different species of territorial birds on Appledore Island. And they all compete with each other because they're all similar. Like they use, they're all usually like shrub nesters and forage for similar things like berries and small insects. And catbirds in particular have two different types of calls. One is the check, which is used for interspecific communication and also sometimes to like warn other birds of like danger or other competitors. And then there's the mew, which is an alarm call and also used for like mate attraction and is also a mimicking, which is why people call it the catbird. Sounds like a cat meowing. So this is all the different check calls of the 4 species of territorial birds I chose on Appledore Island. And these are spectrograms. So it's showing you time in seconds on the bottom and then the frequency on the y-axis. So I'm gonna attempt to play. Okay, oh my God, it's gonna work. That's so exciting. Okay, so that's the catbird call. And then this is gonna be kingbird. And you can hear the frequency is a lot, like the range is a lot bigger. And then the next is red-winged blackbird, which is going to have really fast intervals at a low frequency. And then song sparrows have very consistent high range. Okay. And so possible parameters of how catbirds like discriminate between these different species of bird calls is repetition pattern. So like whether or not this type of call is being repeated once, twice, 3 times, or frequency excursion, which is that frequency range, or interval pattern. And that's high syllable diversity. So it's different from repetition pattern. This is like the time in between each call. And previous research found that repetition pattern was not one of those possible parameters. However, frequency excursion and interval pattern could be. So those are the 2 variables I decided to experiment with. So my hypotheses were that for phase 1, gray catbirds will exhibit a higher degree of responsiveness to conspecific calls than to calls from other territorial birds because their competition is interspecific rather than interspecific. So that's just the distinguishing between different species of calls. And then the second part is phase 2, which is that gray catbirds' discrimination threshold is determined by a call's frequency range and interval pattern. Okay, so my methods for this were going to 4 different locations across the island. The Leighton Porch, Smith's Cove, Devil's Dance Floor, and Founders. And I basically just set up a speaker in a tree nearby where I'd already observed a bird, and then I would play back whatever call I wanted to do. And then I recorded the number of checks, mews, and perch changes 1 minute pre-stimulus as a baseline, and then 1 minute post-stimulus. So here's my first figure. So this is going to be check changes. At the bottom, there are the 4 different call types. And as you can see, it's pretty variable data. But the kingbird data in comparison was significantly less. The responsiveness to the kingbird call was significantly less than to the catbird call. And then I did the same for mew. And again, the kingbird was significantly less compared to the catbird call. All of them were less for other species, but in terms of like statistic significance, the kingbird was. And then same for perch changes. So I decided for phase 2 to choose the kingbird as my other species to modify the frequency range and the interval patterns because I saw that it was the least responded to compared to the catbird call, which was the most responded to. So now what I did to modify frequencies and interval patterns, here is the gray catbird call as a spectrogram. And then I kind of am just showing you a layered kingbird call over that. And then here this is kind of what I did. So I took that catbird call and then I increased the frequency range to match that of the kingbird call. And then for interval pattern, I took the catbird calls and I like copy and pasted it into a new audio file based on how the kingbird interval frequencies were, or interval patterns were. And so this is the first figure I got for just modified frequencies. So just to kind of walk y'all through it, the yellow is going to be frequency type for catbird, and then the blue lines are going to be kingbird frequency range. And then the bottom is the like original call that I'm using. And then I've like modified it. And so from this, I found that for check change, the call type was significant. So in general, they responded less to the kingbird call, no matter the frequency, compared to the catbird, no matter the frequency. And then for mew and perch changes, the frequency type was significant. So as you could see, like whenever it was at a kingbird frequency, it was lower than the responsiveness was lower than if it was at a catbird frequency. And so I started to think about why this could be. Why is there a difference between check and mew? And I was thinking maybe it's because mew is an alarm call, and so it needs to have like a fast threshold, something really quick it can do, like scanning for frequencies. Whereas the check is a communication thing, so they can be a little more specific, which kind of takes me into the next part, which is interval patterns. So for interval patterns, here's my figure. Again, the interval type, catbird interval type is in yellow, and then kingbird in blue. And for this one, for checks, the interval type was significant. So it kind of confirmed what I was thinking about after part 1 of phase 2. And then for mews, there was not really any significance other than call type. And then for perch changes, both call type and interval type were significant. So this could kind of support my idea that checks can be more specific. They can have like a more specific like interval pattern that they're looking for, because it's not putting them in danger of a competitor or like a threat or anything. So after that, my possible parameters were both frequency excursion and interval pattern. And my key takeaways were that catbirds respond more to conspecific calls than to foreign calls, and that frequency range and interval patterns are significant, however, have different effects on the different types of calls. So frequency is going to affect mew calls, and then interval patterns is going to affect check calls. And here are my references. Any questions? My note.
So I understand that you've been on this island for a while, and so I wanted to understand almost your research approach. So why did you choose catbirds? Like, can you talk a little bit about what made you choose this question and this approach?
Yeah, so I took a class earlier this summer, which was field animal behavior, and I met this professor from Duke named Steve Nowicki, really great guy. And he kind of introduced me to like audio and like combining, because I'm also a music major. So combining like that part of my life with like the biology part of my life. And in that class, I did a whole like experiment with song sparrows, doing something really similar about threshold determination across geographic variants. And so I wanted to continue doing that for research in bio. However, like the breeding season is over for most territorial birds here. And so they're not calling as much. However, I noticed just walking around the island that the catbirds were still pretty active. So I sort of just changed gears to a new species, and I still wanted to look at audio. And I had seen that like the catbirds would chase away other catbirds, but were totally fine like hanging out with a kingbird. So I was like, what's going on there? Why are you doing that? Like what's going on? Yeah. So yeah.
Thank you.
Yeah. Morgan.
I think
Okay.
We're like where we got verticals from, the experiment, and where we left on the island, or where they from.
So that's a good question. At first I was gonna record them myself from birds here, and that proved to be very difficult because of the whole issue of sound. But what I did is I just took them from the Cornell Macaulay Library, which has like a bunch of recordings that people have taken of bird sounds. And I did some like research to figure out if there was geographic variance in check calls for catbirds and the other species of birds, and there wasn't. So I found recordings that were all between like late July and early August, so that it was during the period that I was doing this experiment. And they're also all collected from New Hampshire area, so. Anything, is that it? Or is there time for Kelly? Okay, Kelly.
Hey, I just want to clarify, so you, the way you usually, the sound of music, so cool, so you change the frequency and the interval, and then you're like, not the words.
Yeah.
You're not.
Yeah.
So cool.
Okay.
So last but not least for the day, we're going to hear from Emmeline O'Kane, and she will be talking about the effect of ectoparasite trematode Crypticata lingua on killifish, fundulus, heteroclitus behavior, and metabolism. Yeah. All right.
Hi, my name is Emmeline. Okay, so before I get started, I want a quick show of hands. How many of you guys know what a parasite is? Okay, that's good. Now, how many of you know what a tropically transmitted parasite is? Okay, less. So a tropically transmitted parasite uses multiple hosts each at a different trophic level in order to complete its life cycle. And what's interesting about these animals is that they manipulate the behavior of the host. And this can be seen in examples like the Leucochoridium worm and snails. It creates a zombie-like snail, which the parasite infects the eye stalks of the snail, creating a pulsing-like behavior. So essentially makes the eye stalks look like little larvae, and this is more attractive to a bird. It causes the snail to stay out more in the day and up higher on the leaves. And another example, focusing more on what I'm going to be talking about today, is the California killifish, a relative of Fundulus heteroclitus on island. So these have brain-assisting trematodes, and these affect serotonin and dopamine, which essentially increase the swimming behavior of these fish, causing it to be more likely to be eaten by a bird from above. And so kind of like some of these responses you'll see, and specifically Fundulus heteroclitus, they have really good olfaction. So they can sense food and they can tell what is food and what isn't food. And when given 2 bags, they can tell which is food. With other fish, not specifically the killifish, in the presence of parasites, some fish like the stickleback can avoid parasites and avoid infection. They have increased metabolism when they are infected and increased food intake to kind of make up for this intense energetically that the parasite is inflicting on the fish. So with the Crypticata lingua on islands, they are trophically transmitted, like I said, which means they have hosts. In this case, there are 2 hosts, the snail and the fish. So the larva will exit the eggs, enter the snail. They will castrate the snail, reproduce asexually, and the cercaria will then swim out. The cercaria are in green. The cercaria move on to the fish. In this case, are on islands mostly like rock gunnel or some flounder. In this case, there's no sexual maturity happening at this intermediate host, but this is where you can see some sort of alteration of behavior in order to reach the bird, which is the final trophic level, where the parasite reaches sexual maturity, lays its eggs, and then the eggs will then exit in the feces to be reintroduced into this cycle. And what's interesting is that on this island, even though we have this common parasite and common killifish, there has been no studies of like interaction between the two. So that kind of leads me to questioning, how do these killifish respond to these trematodes? And can these killifish sense the presence of these parasites and possibly avoid them? And how is this metabolic rate being affected by the presence of these parasites? To begin with, I had to go and collect my species. I did so using minnow traps, and I went out to Laura Sledge out there. I collected killifish and shrimp. The shrimp will be a food source that I'll go into more detail later. And then I also collected snails, Littoria, and I collected them to get my parasites. So essentially, to test the avoidance part of my hypothesis and questions, I had to make my parasite and non-parasite cues. And I did so by collecting snails with orange feet, as orange-footed snails are more likely to have parasites. And I cracked them open and extracted the cercaria from the soft tissue, which is found closer to the spiral of the snail. And I made kind of a parasite slurry, and I pipetted 2 milliliters of this liquid into a bunch of mini centrifuge tubes, and I froze them to create freezy pops of, of my parasites. And I did this for both the parasite infected and a control, which is a non-parasite freezy pop. And so what I did with these I had behavioral trials. I put my one fish into each bin, and I had 3 trials before exposure, the Q avoidance and the post-exposure trial. The Q avoidance was the only one where I used the parasite Q to sense it, to get a sense of if the killifish can sense the parasites. And you can see the green star is where I put the Q. The blue lightning bolt is where I put the shrimp, the fish, where I put the food, the shrimp, in order to kind of entice my killifish to come out from the rocky area that I put as a shelter. And so to ensure that the fish were exposed, I kind of took them out, put them in a bucket full of infected snails and left them to marinate overnight. And then I continued my trials in the morning. And so what I measured was active time spent non-stationary, the Q interactions, and the number of bites taken. And then moving on to the respirometry part of my experiment, I basically measured the metabolic rate, oxygen consumption over time of both pre-infection and post-infection killifish. And you can see here my little handmade with help of Carrie and a saw. You have a DO probe inside of this container measuring dissolved oxygen concentration over time. And it's in a water bath to also measure temperature and salinity. And I did this 15-minute trials with about 5 minutes of acclimation. And for the behavioral trials, it was about 15 minutes of experimental trials too, with 10 minutes of acclimation in the bin. So really, what did I find? I found that the killifish really cannot sense the parasites. There really is no avoidance. Through a t-test, there's no significant data. They really didn't seem to care at all about there being a little parasite freeze icicle thingy in the bin with them. But with the effect on time, it was sadly also the same with pre, during, and post treatments and the time active. They did not have any significant change of active time. They did decrease their time a bit, but not enough to be significant. But it's interesting, even though they had no effect on active time, maybe it was just like a little decrease, they increased their feeding. So for some reason, all the infected killifish were taking bites out of food after the exposure trial. And this could be because one or two reasons. One is that the parasite is affecting this killifish so much that they have to increase their food intake, like said in the literature. Another one is that simply they're more hungry. Overnight, an extra 10 hours, even though they were starved for around 3 days, maybe 10 hours could have made them even more hungry. So that could also be why they're feeding more. For the respirometry part, we also have non-infected and infected treatments, and then measuring the oxygen consumption over milligrams per liter over minute. And I found out there also was very significant data when doing the t-test, which is interesting because these killifish were eating more post-exposure, except the metabolism slowed. So this is kind of confounding to me because you'd think that eating more, you'd have increased metabolism, but instead it slowed down. So wonder why. Whoops. Okay. So before I get into the discussion, I want to quickly ask, shout out some numbers. How many parasites do you think were found on these killifish? 500. Okay, that's a really great question. Good answers. So I learned a new word from Carrie, oodles. There are so many parasites on here. With more time, I would love to count how many parasites, but you can just see here the little blobs. Maybe it's kind of hard to see, but you can see the little black dots are their eye spots, and they're just everywhere. They're on the eyeballs, and there is even one little one on the gill, which is pretty interesting. And so, recapping with all these parasites, there still seems to be no major change in active time, no major change in they could not sense the parasites and they could not avoid them, but they also had an increase in food intake. And this is interesting because compared to literature, they say the metabolism increases. This could be because we're dealing with ectoparasites in this case, and the other ones in the literature are more brain and cystine or one parasites that infect the internal. And so that could be a reason. And obviously in the future, I would like to control for starvation, maybe have a specific feeding time to see if my data still matches up when they're not being starved. And so respirometry, there is a significant data, and metabolism was confounding. Like I said, if you increase food intake, you expect metabolism to increase as well, but it slowed. So that is some interesting to think about. So in the future, what I would like to do is have, with more time, I can allow these parasites to actually insist in the killifish on fins. and see if this affects their behavior in a different way, if their metabolism changes with now cysts on them. And we can draw more things together about these killifish and these trematodes that don't usually interact on this island. And so I'd like to acknowledge Carrie and Rebecca, the April and Amy from Parasite class for helping me out with some of this stuff too. All of our the Surges, Shoals, some SciComm people, and yeah. Thank you.
Did you say that parents or you saw them in the Heinz and in the field?
Yeah.
Do they have the parasite have a preference or?
Usually the parasites will tend to insist more on the fins, like caudal fins, pectoral fins. But I wanted to see where else it can be found. Some of them have been found on the eyes, impairing vision and kind of like cataracts, which is pretty interesting. The gills was a new one. We don't usually see them on there, but I thought that was interesting too. Wolfgar.
Was in your choice of parasite, was there anything that you wanted beyond that, like other paper? Was it like these are the easiest? Was there any other possibility of a parasite you wanted to find and use, or was like this is the one for me?
Yeah, I specifically chose this one because I knew going into this that these were readily available, and I knew that I could get good data using these parasites. So that's pretty much how I chose that.
How do you think freezing the parasite affected the fish's ability to recognize it?
Yeah, okay, so I kind of forgot to mention this, but when freezing them, the parasites do die. So there is no actual infection being able to happen when I'm running the behavioral trials. But theoretically, even though they're frozen, the fish still may be able to sense it with olfaction or some chemosensory type of signals and cues. I don't know for sure. I would love to do more experiments on how they are able to sense things, but.
All right, so that's it. That's an amazing day. I guess I have the unfortunate task to like try to close this whole day. And as everyone I hope witnessed, there's a lot going on out here during the course of a summer, whether it's a super intense 2 or 4 week experience like the research and biology apprenticeship is, the intertidal crew, they're here for 3 weeks and it probably felt like a month. And then of course our 10-week surge crew that has gone, I don't even know how to explain all that y'all did in 10 weeks. It's a phenomenal amount of work, a tremendous amount of attention to detail, and I feel like everyone has grown tremendously as a result of it. So I'm hugely proud of everyone. I think it also speaks to sort of the scope of research that can happen out here in a very short amount of time. These students have been tasked with a call to be creative, to be engaged, to come up with their own independent research questions. And I think this afternoon proved that they're hugely capable of that. The research topics that were covered, everything from the hagfish at the very bottom of the depths of the ocean, to the intertidal here, to gulls that have been a long-term study topic here. It collectively provides us with a really nice integrated view of all that is going on here in the southern part of the Gulf of Maine. A lot of it focused here on Appledore, but we are very indicative of what's going on around us. As you probably all noticed, a lot of the research questions we're looking at sort of long-term changes and how organisms are reacting to different stimuli. A lot of questions around temperature change and climate change as a result. And a lot of this work is really helping us understand how this ecosystem is responding and reacting to those types of changes. So I, for one, look really forward to seeing all of you move on into whatever direction you take. You've done an amazing job. I'm very proud of you. Thank you for all you've done. Well, it's 3 o'clock, I think. Yeah, 3 o'clock on the nose. That's amazing. So for the rest of the day, for those of us that came out this morning, I fielded an inquiry about whether or not we could maybe stay on island a little bit longer, have a chance to see a little bit more about what our undergraduate researchers sort of experienced during their time here. And so I've talked to Captain Steve, and he's willing to, we're going to switch boats, go back on a slightly faster boat, which will give us a little bit more time on island. So that means that the schedule that you see in front of you is not accurate. We're going to actually look to depart closer to 4:30. So that gives us a nice solid hour and a half. The kitchen, I believe, has prepared some cookies, a little sugary boost to get us through the rest of the day. So a couple of things to do. Please grab a drink, grab a cookie. I believe we're going to open the store for a little while, and then grab a student, take a walk around the island. For those of you that don't have a student to grab and would like to take a walk around the island, I'll be happy to make myself available to show you the facilities. Grab one of our amazing mentors that have guided these students through it all. So yeah, let's meet at the dock at 4:15. Does that work?
Thank you. Thanks for all your help.
Oh, the store's open. My dad has to buy me something.
Get for the rest of the day. RIP photo. RIP photo. Let's call it intertitle photo and a third photo.
Oh, it is.
Hey.
Okay.
Wait a second. All right. Have you couple people?
Yeah, keep going.
Yeah. Are you gonna be here for the file? Okay. I'm doing presentation on the vertical. Yeah. Yeah. So, yeah, like, what is provided. Yeah, I mean, right now, it's, so, we walk out. After you get down that road. Oh, okay, I'll think about that. Yeah, okay, thank you. Yeah. Yeah.